So hello students, welcome back to your own channel Vedantu NEET English. This is Basavaraj sir, your biology master teacher. So students, quickly let me know in the chat right now if I am audible and visible to each and every one of you. Quickly help me know. Because today is going to be one short lecture on the chapter the cell, the unit of life. Now all of you are aware, right? This is not just any random chapter. This is the foundation chapter, and this chapter is the foundation for multiple chapters. That is, if you want to understand entire genetics, this is the chapter, right? So if you learn this chapter, everything in your class 12th is going to be easier on the go. Okay. Now students, I'll tell you one more thing. Who can watch this lecture? This lecture is for all the students who do not have any idea about this chapter. Legit, zero knowledge. You can still watch the lecture. All you need to do is listen in my class, right? All of you, Meghana, Christy, all of you need to just listen in my class and write down the notes which I'm going to give you. You're good to go. Because today is going to be a 3D class. Then what do I mean by 3D? See here. So this is going to be your entire cell. This right here is your entire cell. Now if you look at a cell, students, the entire cell is covered by an envelope. Can you see, students? Entire cell is covered by a membrane, and that membrane is called as cell membrane. Can you see, all of you? The blue color one. Can you see, all of you? That is called as the cell membrane. Okay, cell membrane or plasma membrane. Don't worry, I will show you a 3D visualization of cell membrane also later on. Okay. Now you can see different organelles here. This right here is the centrioles, which helps in cell division. This right here is the Golgi complex right here. See? Can you see Golgi complex? It helps in packaging. Apart from Golgi complex, we have the entire nucleus here, entire nucleus with a lot of chromatin in the center, chromatin. Then we have ribosomes, we have different organelles. So in today's class, I'm going to make sure you understand this chapter by visual representation rather than the boring lecture. Okay. So with that, are you ready for today's class, students? Are you ready, all of you? Can all of you quickly in the chat some fire emoji? With that, let's start today's class. That is going to be the cell, the unit of life. Let's go. Okay, let's go, students. Now, first thing first of all of you. The first thing, right? I want everyone to tell me. I want every one of you to tell me what is the common thing in a bacteria, a plant, an animal, a fungus, or any type of organisms. Every single living organism has that one thing in common, students. All of you look around. Tell me, what do you see? For example, if I look around myself right now, I will see light, I will see camera, I will see a pen, I will see notebook, I will see my mobile phone. I will also see certain different types of ants walking around. I will see. In the night time, I will see a cockroach. So what is the difference between all of this cockroach, ant, spider, human with respect to a pen, book, paper? What do you think is the difference, students? What do you think is the difference between all of them? It is none other than the cell. It is none other than the cell, students. It is none other than the cell. It is the same cell, students. It is the same cell. It is the same cell which is found in. It is the same cell which is found in all the living organisms. All the living organisms. Okay. Now, students, you tell me in the chat, all of you, do you find the cells in the non-living things? Do you think this pen right here, is it made up of cells? No. So non-living things will not have any cells. Non-living or inanimate things, right? There is no cell. There is absolutely, cell is absent. Now I'll tell you two scenarios. Okay. There are certain organisms in this world who are made up of a single cell. Yes, a single cell. But there are us. We humans are made up of multiple cells. So when this cell right here, the living organisms can be of two types. one type of organism is made up of single cell. Such organisms are called as unicellular organisms. It is called as unicellular organisms. What about us? We are multicellular organisms. We are multicellular organisms. Right? Is that clear to all of you? Is that clear to all of you? Unicellular means single cell. Multicellular means many cells. Means many cells. Okay? Many cells. Now students, let's understand a little bit of discovery and the history right now. Small history. Now who do you think discovered the first living cell, students? Who do you think discovered the first living cell? Anyone in the chat who can tell me? In fact, the first living cell was not discovered by Robert Hooke. This right here is Robert Hooke. This right here is none other than Robert Hooke. Robert Hooke. Now this Robert Hooke did not discover the living cell. Robert Hooke observed the cork cells and he discovered the dead cell. Dead cell. Then who do you think observed the living cell then? Living cell. The first living cell was discovered by none other than the father of microscopy. Can anyone tell me who is known as the father of microscopy? The father of microscopy is none other than the Antony von Leeuwenhoek. He was a scientist who observed the living cell. Living cell. He observed the living cell. Now this living cell, dead cell. Okay. Now I'm telling cell from that time. But what exactly is a cell? What exactly is a cell then, students? What exactly is a cell? Now just like how if you look at a building, every single building is made up of bricks by bricks, right? Our entire body is also made up of small bricks. Small bricks. And these small bricks are called as this small bricks, right? Like how a building is made of small bricks like this. Our entire body is also made up of this small bricks, and these small bricks are called as cells. Now there is a famous definition for a cell. Can anyone remind me the famous definition? There's a famous definition, right, for a cell. The famous definition of a cell is called as, it is the basic unit of life. It is the basic unit of life. Now what if I tell you one more definition now? What is a cell? The cells, this is the better definition. What is the better definition, students? The cells are the smallest. They are the smallest structure. They are the smallest structure that are capable. They are capable, okay, of independent existence. Independent existence. This is the definition which I want everyone to remember. What is a cell, students? Cell is the smallest structure. Cell is a smallest structure which is capable of independent existence. It can survive on its own. Now I'll give you a simple example for unicellular organisms. For unicellular organisms, what is the case? Okay. For unicellular organisms. For unicellular organisms. Okay. For unicellular organisms, cells are independent. They will give independent existence, as well as they will perform all essential function. They'll perform all essential function. Just remember these 2 points. Remember these 2 points, students. That is, in the case of unicellular organisms, it will show independent existence, a cell, and will perform all the essential functions of life. Okay. Now that is clear to all of you. Now all of you know just simple definition of a cell. This is the basic definition of a cell. Better definition. All of you understood that living cell was discovered by Antony von Leeuwenhoek. Dead cell was discovered by Robert Hooke. Okay. Now a question to all of you. Who do you think discovered the nucleus? Because if you look inside a cell, it could be a plant cell or an animal cell. For example, students, if you do not know what is a nucleus, let me show you. Let me show you, students. Let me show you a nucleus. Never mind. Students, if you see a typical plant cell or animal cell, a typical plant cell or animal cell, in this entire cell, right, there is something called as a brain of a cell. We call this as a brain of a cell, where all the genetic information is stored. For example, this is a plant cell right here, right, and if you see here, right, this is called as the brain of the cell right here, brain of the cell, and this here is the nucleus. Now the question to all of you, a question to all of you, who discovered the brain of the cell, also called as the nucleus? Anyone? The nucleus was discovered by, students, nucleus was discovered by Robert Brown. Robert Brown was a scientist who discovered the nucleus. Now you might ask me, students, in which organism, sir? Where did he observe the nucleus? Did he observe in human cell? Did we observe in the case of an animal cell or a plant cell? He observed the nucleus in a plant cell, that is, he observed in the orchid roots. A cell of the orchid roots, he observed it. Okay, that is Robert Brown. Now, apart from all this, students, now we come to one more concept called as cell theory. Right? What exactly is cell theory, and what does cell theory tells you, and who are the scientists involved in the cell theory? Can we start? All of you? Can we start, students, now? Cell theory. Now, students, I want you to write down these notes. Today, I'm going to make you write down a lot of notes. So, write down these notes. Who discovered living cell? Who discovered dead cell? Who discovered nucleus? Okay, one by one, I want you to write down this. Okay. Now, understand the cell theory. Now, cell theory basically tells you that what is cell made up of? That is, what are we made up of? Okay. Now, students, understand. This entire cell theory was given by, initially it was given by two scientists. Initially, cell theory was given by two scientists. Can anyone name them if you know them? Right. In 1883, students, there was a scientist. He was a German botanist. He was a German botanist. Okay, he was a German botanist, the first scientist. Remember, students, write down this information, will be very useful to all of you. His name was Matthias Scheiden. His name was Matthias Scheiden. Now remember, students, remember this name is very important. They can ask you, German botanist, who was German botanist? Directly they can ask you. Okay, Matthias Scheiden. Now, since he was a botanist, students, do you think he'll work on plants or do you think he'll work on animals? Obviously, he's a botanist. So, his main work was on plants. Now, understand very carefully. When he was working on the plants, when Scheiden was working on the plants, he noticed that plants are made up of different type of cells and different types of tissue. That's all. Okay. So, plants are made up of different kind of cells. Okay. Done, students? Done? Now, in the year 1839, there was one more scientist. Now, he was not from the German. He was from Britain. He was a British zoologist. Okay, he was a British zoologist. His name was, students, anyone can tell me who was his name? His name was Theodore. Very fancy name. Theodore Swan. His name was Theodore Swan. Theodor Swan. Now, unlike your Mathers, who was a botanist, he was a zoologist. So obviously he will work on animals. Yes, he worked on animals. When he was working on animals, he observed a particular structure around the cell, which is protective in nature. Today we know it as cell membrane. As cell membrane or plasma membrane. He also noticed that, students, even in plant cell, in the case of plant cell, there is an extra covering which is missing in animal cell. Can anyone tell me what is that? There is an extra protective layer around the plant cell which is missing in animal cell. He called it cell wall. That's the cell wall. So cell wall is present only in the case of plants. Only in the case of plants. And the third thing, he told us that students, animals like us and plants, students, all of them, animals and plants, they are made up of cells and product of cells. This is very important. So he told us that all the animals and plants are made up of cells and a product of the cells. Now came one more confusion. What is one more confusion, students? The one more confusion was that these two scientists, right, they could not explain how did the cells came into existence. Like, for example, that is all right. I am made up of cell and product of the cell. But where are the cells originating from? Now, this information was given by one more scientist called as Rudolf Virchow. Okay, there came one more scientist, students. There came one more scientist called as Rudolf Virchow. All of you remember? See, this is the Mathis Scheiden. This is Theodor Swan. Too handsome. All of you, namaskara. Quick namaskara you give. Yes, done. This is called as Rudolf Virchow. Rudolf Virchow in 1855. Students, years are also important. In the year 1855, Rudolf Virchow came and told, wait a minute, you are not able to tell. Doesn't mean I cannot also tell. So Rudolf Virchow came and told us that cells originate from cells divide, okay, cells divided and new cells are formed from pre-existing cells. So he told us that all the new cells, when the cells divide, right, all the new cells are arising from where? From, this is a very important line, from pre-existing cells. So all the cells are arising from the pre-existing cells. Now here he given famous Harry Potter ka, what is that one magic mantra I gave. What is the magic mantra? The magic mantra is omnius cellula e cellula. Omnius cellula e cellula. Okay, it means that cells arise from a pre-existing cells. Now this is all right. Now this last line, the first one line, that is cells arise from pre-existing cells, as well as animals and plants are made up of cells and product of the cell. So this line and that line together, it is called as a cell theory. And the cell theory was given by three main scientists, that is German botanist Mathis Scheiden, British zoologist Theodor Swan, as well as the Rudolf Virchow. All the three scientists together gave the cell theory. Is that point clear to all of you? Now a question arises, that is there any organism? Is there any organism which does not follow cell theory? Anyone? I'm looking at the chat right now. I'm looking at the chat. Can anyone tell me the answer? Any organism which does not follow the cell theory. Is there any organism? Yes, there is. The famous celebrity known as the virus. Known as the virus. So virus is an exception to the cell theory. Remember that. Virus is an exception to the cell theory. Okay. Now, with that, we come to the next concept of today called as cell overview. Now, I believe till now everything is clear to all of you. If it is clear to all of you, I want everyone to quickly smash that like button right now because it helps in YouTube algorithm. Honestly, it does. Okay. And also some fire emoji in the chat to let me know that there is no doubt till now. Quickly, all of you. Quickly, all of you. Yes. All of you. All of you students. Quickly. All of you. Quickly. Fire emoji in the chat. Now we come to something called as cell overview. Now in cell overview, I will tell you what exactly have we learned till now. Right? What exactly have we learned till now? We have learned that there is something called as a animal cell. Okay, there is something called as a animal cell. There is something called as a plant cell. And in this plant cell, we have the cell wall. Cell wall is present in the plant cell. Okay. Now, there is something called as nucleus. Nucleus. Within this nucleus, there is the DNA. That is, genetic material is present inside the nucleus. It could be DNA or RNA in some cases. RNA in some cases. DNA or RNA. Genetic material. Okay. Now students, in cell overview, I want you to understand the entire cell can be broadly classified into two categories. Not animal and not plant. I'm not talking about animal plant. I am talking about a classification called as, does anyone know what I'm talking about? Basic classification called as, based on the membrane bound organelle. Based on the membrane bound organelle, the cells can be broadly classified into prokaryotes as well as eukaryotes. Now, what is the difference? What is the difference between prokaryotes and eukaryotes? Very simple difference, students. In the case of prokaryotes, these cells, there is no membrane bound. No membrane bound. The membrane is completely absent here. That is, no membrane bound organelles. That is, there is no extra covering around the membrane in the case of prokaryotes. Okay, prokaryotic cell. Okay. Now, what about eukaryotic cell? Eukaryotic cell are membrane bound organelles. They are the membrane bound organelles. Membrane bound organelles. Okay. They have membrane bound organelles. Okay, membrane bound organelles. Every single membrane, it could be endoplasmic reticulum, Golgi complex, nucleus, right, chloroplast. Every single membrane has a outer covering around it. Outer covering around it. Clear? Clear. Now, students, understand here. Here, I told you just now, there is endoplasmic reticulum. There is ER here, ER, Golgi complex, nucleus, mitochondria, right? Mitochondria. All of these organelles have a separate layer around it. Now, let me ask you a very simple thing. Okay. Now, do you think under membrane bound organelles, that is eukaryotes, are there any exception? Are there any organelles which do not have any membrane? Yes, there is. Yes, there is. In fact, there are multiple. Here, there are two main categories. Two main categories. One is found in the case of animal cell. One is found in the case of plant cell. Okay. The first one is called as ribosomes. It is called as ribosome. And let me tell you, students, let me tell you, listen to me very carefully. First one is called as ribosomes. Now, what if I tell you the same ribosome is also found in the case of in the case of prokaryotic cells. So remember, ribosome is one of the organelles, not the only one. It's one of those organelles which is present in the case of prokaryotic as well as eukaryotic. Now you think logically. If it is present in the case of prokaryotic, means no membrane bound, so no membrane bound. Okay. Now other exception is going to be found in the case of only animal cell, and that organelle is called as centrosome. It is called as what? Centrosome. Students, remember, this is the same centrosome. This is the same centrosome which helps in cell division. Clear, students? Now you might be wondering, sir, here also have ribosome, here also have ribosomes. Are they same? Do you think we have the same ribosome in the case of prokaryote as well as eukaryote? Do you think that? The answer is no. In fact, the ribosome present here is called as 70S ribosome. 70S. Now, what about here? The ribosome in the case of eukaryote is called as 80S ribosome. 80S. So, even though they might be present in the case of eukaryote as well as prokaryote, they are not same. They are absolutely not same. One is 70S, one is 80S ribosome. Is that point clear to all of you? Is that point clear to all of you, students? Yes? Yes? Okay. Now, I hope that is clear to all of you. Now we go to the next concept called as cell size. Obviously, there are multiple different size of the cell. All the cells are not the same size. They're not the same size. Some cells are going to be very large, some cells are going to be very small. Yeah. Some cells are going to be very long, some cells are going to be very short. Okay. Remember that. Now, first thing, first of all, which is the smallest cell? Smallest cell. Smallest size. Largest. Longest. What about in the case of bacteria? What about in the case of a human RBC cell? Human RBC cell. What are the different sizes? Now here you need to remember the numerical value also. You have to remember at any cost. It is for sure. They will ask you. Okay. Now for example, the smallest cell. Smallest cell is nothing but the mycoplasma. Is going to be the mycoplasma. Now this mycoplasma is around 0.3 micrometer. Is micrometer. Okay. The largest cell is going to be the egg. Egg? Yes. Egg of ostrich. Egg of ostrich. Okay. Now what about bacterial cell? What do you think the size of the bacterial cell? The size of the bacterial cell is relatively larger compared to a mycoplasma. A little bit more than that. Okay, larger than that. It is around 3 to 5 micrometer. 3 to 5 micrometer. Done. Now what about bacterial cell? Do you think bacterial cell? What's the longest? One second. Bacteria. Longest is going to be nerve cell. Nerve cell. Now human RBC. Human RBC is around 7 micrometer. 7 micrometer. Okay. Now one question arises. One famous question is there, students. One famous question is there. Now this is the mesh. Here I have a mesh. Okay. Here I have a mesh. Now in this mesh, the size of this mesh is around, let's say, 0.1 micrometer. 0.1 micrometer. Do you think bacteria can pass through it? Tell me. Do you think bacteria can pass through it? Absolutely not. Do you think mycoplasma can pass through it? No. But do you think a virus can pass through it? The answer is yes. Because the virus is around 0.001 micrometer. So it's even more comparatively tiny, very small compared to a bacteria. So a virus can pass through, but not a bacteria, not a mycoplasma. Okay. Now here there is a famous question in your biology classification, that is mycoplasma lacks cell wall. The cell wall is absent here, remember that. They can ask you the question anywhere. Okay. They can ask you this question. Okay. No. So RBC cannot pass through, bacteria cannot pass through, mycoplasma cannot pass through. Okay. Now students, here are the different shapes they have given you. Red blood cells which are round or biconcave in nature. Then we have white blood cells which are amoeboid in shape. Columnar epithelial cells which are elongated, narrow. Then there is a nerve cell, I told you, longest and the branched. Then there is a tracheid. Tell me quickly in the chat, students, is this tracheid as part of xylem or phloem in the chat? Then we have mesophyll cell here, mesophyll cell. Mesophyll cells are the photosynthetic cells which have the chlorophyll. Mesophyll cell, round oval shape. Now here there is size. Eukaryotic cell 10 to 20. Virus, see, 0.02. Size can be very tiny viruses. Mycoplasma PPLO, that PPLO 0.1. A typical bacteria is around 1 to 2 micrometer. Okay, micrometer. Now students, now we come to the entire prokaryotic cell. Till now the basic foundation is laid out. Entire basics is over. Because students, let me tell you, this is going to be a very detailed class. I'm going to make you write the information. You will write the information, you will learn it and take away today only. And after today's class, there has to be no backlog whatsoever in this chapter. Clear? Clear all of you? Yes? Now, can we start? Can we start the prokaryotic cell now? Prokaryotic cell. Can we start? All of you? Okay. Nuclei is the part, basically nucleus, right? Organelles are the different structures found here. Okay, organelles are different structures. Now, part of xylem. Okay. Prokaryotic cell. Now the first question arises, this prokaryotic cell, how does it look like? How does it look like a prokaryotic cell? And in which kingdom do you find prokaryotic cell in a majority? Okay. Now the first question I'll answer, that is, prokaryotic cell can be mainly found in the kingdom Monera. In the kingdom Monera. In the kingdom Monera. Now second question, how does it look like? How does the prokaryotic cells look like? Does it look like a RBC? No, it does not. Let me show you. Okay. This is how a prokaryotic cell looks like, students. This is how a prokaryotic cell looks like. Can all of you see this? This is a typical prokaryotic cell. I hope it is visible to all of you at this point. Can you see? This is a typical bacterial or a prokaryotic cell. It looks like one rat, no? The rat it is. Okay. Now if you see here, in this prokaryotic cell, there is no membrane bound organelles. Membrane bound organelles are completely missing here. Okay. Now if you see here, it has a long structure for motility. This right here is called as flagella. Flagella helps in motility here, motility. Okay. Now students, apart from that, students, can you see pili here? Pili is the surface, on the surface, pili and fimbriae, two structures. Pili and fimbriae, their main function is attachment. Their main function is what? Attachment. Flagella will help in motility, pili will help in attachment. Okay, attachment. This is the entire capsule. Now what is capsule here? Capsule is actually part of glycocalyx, that is the outermost layer. Don't worry, I'll teach you today. What is capsule? What is slime layer? All that I'll teach you. Capsule is basically thick and tough glycocalyx. Okay. Now here, if you see here, students, there is cell wall here. Okay. So right now, all of you understood that there is an outer layer. Inside that, there is a second layer is called as a cell wall. Second layer is called as the cell wall. So if someone asks you from today onwards, do you find cell wall in the case of bacteria, in the case of prokaryotic cell? What is your answer? Yes. So it is not only the plants which have the cell wall is also present in the case of bacteria. Remember that. Okay. Now what is the innermost layer? Is the plasma membrane. Now students, can you see here? Can you see here? Is there any specific location as nucleus here? Do you find nucleus structure here, students? Can anyone tell me? Absolutely not. There is no structure called as nucleus. In fact, the DNA is scattered, and DNA is scattered in the form of nucleoid. Okay. Apart from that, there is the cytoplasm here. Cytoplasm. The cytoplasm is present in the case of prokaryotes and eukaryotes, and this right here is the ribosomes. Here, a few things are missing, such as mesosomes, that I will draw and show you. Now, all of you have a clear idea in your mind what exactly is a prokaryotic cell, students? All of you agree with me? All of you have a clear idea in your head now? Yes or no? Tell me. Yes or no? All of you have some clear idea? Yes. No, students. No. With that clear idea, now we enter the prokaryotic cell. With that basic information back of your head, with some visualization, we can start prokaryotic cell now. Okay. Now the first thing which comes up is in which organisms do you find prokaryotic cell? Obviously, the first answer is in the case of bacteria. You will find here bacteria. In the case of blue-green algae, okay. In the case of blue-green algae, in the case of mycoplasma, in the case of PPLO. So all of these organisms, in all of these organisms, we can find the prokaryotic cell. Okay, prokaryotic cell. And remember, students, compared to eukaryotic cell, prokaryotic cell is actually very small and tiny, and it can multiply very fast. That is, prokaryotic cell is always smaller and it can multiply very fast. Okay. Now, what are different shapes here? There are different shapes of prokaryotic cell. Some of them can be 𝐂𝐤. 𝐂𝐤 means spherical in shape. Some of them can be rod shape. This is rod shape, means bacillus. Some of them can be in comma shape. Yes, comma shape. That is called as vibrio, comma shape. Some of them can be spiral shape. The shapes of prokaryotic cell can vary. It can vary depending upon different organisms. Prokaryotic cell. Okay. Now this is a prokaryotic cell which I showed you beforehand. Can all of you see again? Outer layer, middle layer, inner layer, and everything. Now students, let's draw this. All of you. I want everyone to draw with me. Open your books and start drawing. All of you, open your books and start drawing. Okay. This is the outermost layer. Done. Then let's do a second layer now. This is the second layer. Okay. This is the second layer. Done. Now let's do the innermost layer. This is the innermost layer. Can all of you see this? This is going to be the innermost layer. Done. Now students, here, what is the outermost layer called as? The outermost layer is called as glycocalyx. The outermost layer is called as glycocalyx. Now here I want everyone to remember that this glycocalyx can be present in two different forms. This glycocalyx can either be present in the form of loose sheath, or it can be found in the form of thick and tough layer. Now when it is present in the case of loose sheet, it is called as slime layer. it is called as slime layer. When it is present as thick and tough, it is called as capsule. It is called as capsule. Done. Now what is the second layer? The second layer is called as a cell wall. It's called as a cell wall. Now I will ask you one more question. Now what is the cell wall made up of? This cell wall is made up of a substance called as peptidoglycan. Peptidoglycan. Now what is this peptidoglycan made up of? This peptidoglycan right here is made up of two molecules called as NAG and NAM. N-acetyl muramic acid, N-acetyl glutamic acid. NAG and NAM. Okay, NAG and NAM. Now, what is the innermost layer? The innermost layer is called as the cell membrane. It's called as cell membrane. Innermost layer is called as the cell membrane or plasma membrane. And this cell membrane or plasma membrane is selectively permeable. It is going to be selectively permeable. It means that it will allow only certain substances in and out of the cell. Not everything is allowed to go out and in. Okay. Now students, here for motility, for motility, there is a long structure called as what, students? Which color should I use? Different color. There is a long structure called as what, students? Can anyone tell me? What is this long structure for motility? That is called as flagella. This right here is called as flagella. It's called as flagella. Now, apart from the flagella, do you remember there was a structure which was helping in attachment? There was a structure which was helping in attachment. What do you call that, students? Structure which was helping in attachment. This was called as pilae or fimbriae. Or fimbriae. Clear to all of you? Clear. Now we have covered the most of the outer part. Now we go to the inner part. Now what do you think this inner part is made up of? This inner part is made up of a simple substance called as cytoplasm. This inner part is made up of cytoplasm. So there is inner part is called as cytoplasm. Cytoplasm. Okay. Now apart from the cytoplasm here, if you notice from the membrane, from the inner membrane of cell, inner membrane of your entire bacteria or the cell here, that is from the cell membrane, there are certain extensions in this. Can you see, all of you? There are certain extensions here. There are certain extensions here. Extension of cell membrane. These extensions are called as mesosome. These extensions are called as mesosome. They are called as mesosome. Okay, mesosome. Clear, students? Mesosomes. Now you might be wondering, sir, what is this function of mesosome? That I'll tell you later on. Give me 2 minutes. What is the function of mesosome? Now what are we missing? Now here, students, similar to mesosome, there is one more extension here. Similar to mesosome, there is one more extension here. But this extension here, right? This extension here has certain pigment. This extension right here has certain pigment. Now this extension right here is called as chromatophore. It is called as chromatophore. Called as chromatophore. Can all of you see this chromatophore? This is the pigment causing. Now the same chromatograph can be found in the case of blue-green algae. In blue-green algae. It can be found in blue-green algae. Now, apart from that, there will be obviously there is going to be the 70s ribosome. There is going to be ribosome. That is 70s ribosome is going to be there. 70s ribosome is going to be there. Now, where is genetic material? Now here, genetic material is going to be scattered. Genetic material is going to be completely scattered here. That is called as nucleoid. Here, genetic material is called as nucleoid. It's called as nucleoid. That is the entire genetic material here. Entire genetic material. Now that is your entire prokaryotic cell. That is the entire prokaryotic cell. Students, can I give you a small recap of this entire thing? Can I give you an entire small recap of this entire thing one more time? One more small recap of this entire thing. A small recap. Students, is everyone following? No. Let's start with the first thing. Let's have the first one. That is the outermost layer. The outermost layer is called as glycocalyx, loose sheet, thick layer. Loose sheet, slime layer, thick layer is called as capsule. Second layer is called as a cell wall, made up of peptidoglycan, NAG and NAM. That is basically type of heteropolysaccharide. If you remember biomolecules, heteropolysaccharide. Then there is cell membrane, which is a lipid bilayer here also, slightly permeable. Flagella for motility, fimbriae and pilae for attachment. For attachment. Then there is nuclear genetic material. There is completely no nuclear envelope. Nuclear envelope is completely missing here. Envelope is missing. Then there is mesosome. Mesosomes basically are the extension of plasma membrane which help in respiration. There are multiple functions. I'll make you write down. Then there is chromatophore. Chromatophore main function is what? Provides pigment for blue-green algae. That's all. Ribosomes is there here. Okay. I believe we have covered almost every single structure here. I will move out. If you want, you can take a screenshot of this. If you want, you can take a screenshot of this, students. All of you take a screenshot of this. Done? Done, students? Done? All of you? If this is done now, students, now I'll teach you what exactly is mesosomes. Remember this mesosome. This mesosome is very, very important for the cell here, because this mesosome has multiple functions. One by one, write down, because there has been a NEET PYQ on this question previously on this mesosome. Okay. Now before we go, remember mesosome is what, students? It is an extension. It is an extension of cell membrane. It is an extension of cell membrane. Now this mesosome can be found in multiple different forms also actually. It can be in the form of vesicles, like round it can be. In the form of vesicles it can be. Okay. It can be tubular also. It can be tubular like this, or it can be in the form of lamellae, something like this. So it can be found in multiple different shapes. It can be found in multiple different shapes. Okay. It's tubular shape. Okay, tubular shape. Now, what are the functions you write down? What are the functions? This is very important. From here, the questions will come. What are the different functions? Write down all of you. Okay, write down functions. First one is they help in cell wall formation. This is the first point only they have asked multiple times. They help in cell wall formation. They also help in DNA replication. DNA replication. Students, write down this one. They will ask you multiple times, they have asked. Third thing is they help in respiration. They help in respiration. Fourth point, they help in secretion process. Fifth point, they will increase the surface area. They increase the surface area. Okay. Sixth point, they have certain enzymatic content, or they help in enzymatic reaction actually. Okay. Last seventh one, they help in distribution of daughter cell. Distribution of daughter cells. Distribution of daughter cells. Now they will ask you any one of the following, and they'll tell you which of the following is not a function or which of the following is a function of mesosomes. You should be able to tell this much. Clear, all of you? Clear? Extension of plasma membrane or cell membrane, different shapes, functions. Clear to all of you? Clear to all of you, students? All of you wrote down? All of you wrote down, students? Make these notes. All of you, if you are able to make these notes right, trust me, all you need to do is revise these notes and you're good to go. You don't have to go back to the books again and again. Do not read the textbook again and again. Okay. Now, students, if you look at this entire envelope of the plants, of the bacterial cell or the prokaryotic cell, there is something called as a peptidoglycan layer. All of you remember, right? In the cell wall. In this cell wall, there is a peptidoglycan layer, right? Now, students, remember this. There is something called as gram-positive bacteria and gram-negative bacteria. Now, what is the use of this? Now, students, if you know, there is something called as classification, taxonomical classification. But did you know there is a taxonomical classification even for bacteria? Yes, there is. There is a taxonomical classification for bacteria also. And the first step in classification, or the most basic level of classification is gram-positive and gram-negative bacteria. Now, what is the difference? Difference is the layer of peptidoglycan. If you see the cell wall in gram-positive, large layer, gram-negative, small layer of peptidoglycan. Now, because this small layer, students, because of this small layer of peptidoglycan, this gram-negative bacteria will look under the microscope around red color, and this one will look violet color. Now, why is that? The answer is very simple. Okay, if you want me to explain, I'll explain in a very small manner. When we add alcohol in the gram staining, right, these holes here, the lipid layer is very less. Okay, lipid layer is very less. The lipid layer will open up, all the stain is going out, the first stain is going out. But here, the first stain is retained here. Crystal violet is retained here, crystal violet is not retained here. And here, saffron is retained in the last step, saffron. That is why it looks almost reddish pink color. Here it looks violet color. Okay, violet color. Okay. Now, students, now we come to flagella. All of you remember flagella? Yes. Flagella helps in what? Basically, bacteria are mainly of two types. All of you remember? Bacterial cells are of two types. Some of them are motile, some of them are non-motile. Okay. Now, these motile bacteria have something called as flagella. Okay. Now, this flagella has three main parts. That is, it has a structure called as filament on top, it has a hook in the middle, and it has a basal body. Students, let me show you the diagram. For this. It will be easy to remember or visualize also. See here, all of you. This is the basal body, the middle basal body. This is the entire hook, and this is the filament. One more diagram I'll show you. Can you see here? This entire thing is the basal body. This is the hook here, and this is the filament here in the flagella. Now, what you see here, right? This long structure, that is basically the filament part. So, filament, hook, and basal body. Three main parts of the flagella. In the case of prokaryotes, basal body, hook, and filament. Three main steps, three main parts. Clear, all of you? Clear? No. Next is ribosomes. Now, I already mentioned ribosomes are the structures which are responsible for protein synthesis. You might be aware, right? If you know the chapter molecular basis, ribosomes are the structures which help in protein synthesis. Yes or no? Yes. Now, here in the case of prokaryotes, the ribosomes present here is 70s ribosome. Now, this 70s ribosome can be divided into 50s and 30s. 50s and 30s. But what is 50 plus 30, students, tell me mathematically? 50, 60, 70, 80. It is supposed to be 80, right? 50 plus 30 is 80. But why do you say 70 here? The answer is very simple. Now here, this is not based on mathematical addition. It is based on the sedimentation coefficient or based on the sedimentation property. Now what do I mean by that? I'll give you a simple explanation. Listen to me very carefully, all of you. Now if you take this entire ribosome, okay, let's take three different ones. Let's take one entire 70, let's take one 50, and let's take one 30. Okay, two subunits and an entire full. You keep it into a test tube. Or F&op tube. Okay, and make it undergo centrifugation. Nicely undergo centrifugation. And after some time you will notice that the entire 70 subunit is somewhere here, 50 subunit is somewhere here, and 30 subunit is somewhere here. That is, based on the density, they are separating out. Okay, so we have a measuring scale here also, measuring scale. So based on the density, we are classifying them. That is, 70 is made up of 50 and 30, not because of addition. Clear, all of you? Yes. Now here, students, what is this S? From that time I'm speaking, I'm telling you. What is this S? This S can be called as sedimentation coefficient. Okay, or this S is also called as Sedberg unit. It is also called as Sedberg unit. This is the same as Sedberg unit or sedimentation coefficient. That is the ribosome here. Now students, one small concept is here. Okay, one small concept before we go to next concept. Okay, that is called as polyribosome. Okay, now what happens is sometimes in this bacteria and all. Okay, for example, this is one mRNA. Let's just say this is one mRNA. Okay, on this mRNA there are multiple ribosomes. Multiple ribosomes are there on this mRNA. That is several ribosomes plus mRNA, a single mRNA. This is called as polyribosome. It's called as polyribosome or polysome. So several ribosomes on a mRNA is called as polyribosome or polysome. Very simple. Now next concept is called as inclusion bodies. What are these inclusion bodies and what is the use of them? Okay, think of inclusion bodies as the storage unit. They are the typical storage unit found in the case of prokaryotic cell. Their main function is only storage. They will hold things. Okay, they're basically reserved material. Or storage unit. Now here are a few examples are there given you in NCERT. That is different types of storage units. Okay, different types of storage units are there. Okay, like phosphate granules. Are there. Then there is cyanophysin granules. Synophysin granules. Then there is glycogen granules. Now students, there is something called as also gas vacuole. Gas vacuole. Now the surprising part is this gas vacuole is only found. This gas vacuole is only found in purple and green sulfur bacteria. In purple, blue, green, and what do I write, students? And purple green bacteria. Okay, so these are different types of inclusion bodies. Can you see here? All of these are different inclusion bodies inside the cell. All of these inclusion bodies will hold things. They will store things inside them. Store things inside them. Clear, all of you? Clear? So with that, prokaryotic cell is over. Now students, I'll give you exactly 5 minutes, not 5 minutes, I'll give you exactly 15 seconds to ask your doubts, because I'll be looking at the comments now. Okay, I'll give you exactly 15 seconds or 20 seconds, 20 seconds and ask your doubts. All of you? Yes? Exactly. Okay. All of you who are new to the channel, quickly smash the like button. Yes? All clear? What exactly 70S? 70S is the ribosome name. This entire thing is called as 70S ribosome. The meaning of S is Sedberg unit. This entire thing is called as 70S ribosome. Okay, 70S ribosome. Okay, this is where proteins are produced. This you will learn in molecular basis, right? You will learn in molecular basis. There will be small subunit will come, large subunit will come. In the middle, there will be mRNA. Subunit mRNA will move, and protein synthesis will happen. Biomoles are already taken. Okay. Now students, let's start with eukaryotic cell. This is polyribosome, done. Now let's start with eukaryotic cell. Shall we? Let me take some water. Students. Now students, prokaryotic cell, right? Prokaryotic cell was found in kingdom Monera, in the case of kingdom Monera. Now where do you think eukaryotic cell can be found? Do you find eukaryotic cell in the case of Protista? Yes. Do you find eukaryotic cell in the case of kingdom Plantae, Animalia, Fungi? The answer is yes. The first point is very simple to all of you. In where do you find? Eukaryotic cell can be found in the case of Protista. It can be found in the case of Plantae, in the case of plants, in the case of animals, and also in the case of fungi. So this is the different kingdoms where the eukaryotic cell can be found. Now students, remember, unlike prokaryotic cell, eukaryotic cell, all the members are membrane bound here. All the organelles are membrane bound. All the organelles are membrane bound here. Are going to be membrane bound. They are going to be membrane bound. Now here there will be a proper nucleus. We can find a proper nucleus, and inside the nucleus there is going to be DNA, and DNA is going to be in the form of chromosomes. In the form of chromosomes. Clear, all of you? Clear? Now here students, there is going to be something called as cytoskeleton also here. Cytoskeleton. We'll come back to this because there is going to be a complex locomotory things which will help in locomotion here, because there is something called as cilia and flagella here. Cilia and flagella. Cilia was absent in the case of prokaryotes. Right, here we will have cilia and flagella will be present here. Okay. Now, next thing is going to be plant cell versus animal cell. Now, students, quickly tell me in the chat, students, which is that one organelle, outer organelle, which is present in the case of plant cell and absent in the case of animal cell. Any one of you tell me. The first organelle I'm talking about is cell wall. Is going to be cell wall. So cell wall is going to be present here and cell wall is going to be absent here. It is going to be absent here. Now what about plastids? Plastids is going to be present here. It is going to be absent here. It is going to be absent here. Okay. What about centrioles? Centrioles is going to be present here, but it is going to be absent here. Okay. So this basic difference you should know. For example, if I give you this diagram right now, tell me which one is plant cell and which one is animal cell. Let's just say this is A and this is B. Tell me which one is an animal cell, which one is a plant cell. Go. Tell me. Which one is a plant cell, which one is an animal cell? If you see here in the diagram of A, can we see a structure called as chloroplast here? Chloroplast. Okay. Can we see cell wall here? Cell wall here. In B, we can find something called as centrioles here. Centrioles. Okay. Cell wall is absent here. Now, in the case of A, can we see a large central vacuole? Can we see here? Large central vacuole. Large central vacuole. So, A is going to be what, students? A is going to be a typical plant cell and B is going to be a typical animal cell. So, plant cell and an animal cell. Simple. Okay. Now here, many of you find it difficult to understand the cell membrane. Yes or no? Tell me, all of you. Many of you find it difficult to understand the cell membrane because you don't visualize it. Because now I'm going to teach you in 3D right now. I'm going to make sure you understand what exactly is the cell membrane, how does it look like, and how does it function. Can we start? All of you, can we start now? What exactly is cell membrane? Okay. Now, before we go further, what is actually cell membrane made up of? What is it made up of, students? It is made up of lipids. All of you agree with me? Because we tell cell membrane as a lipid bilayer. So it has to be made of lipids. It is made up of lipids. Now, apart from lipids, it is made up of certain proteins are there, intrinsic and extrinsic proteins. There are certain carbohydrates are there. Okay. There are certain carbohydrates. Apart from carbohydrates, there are other certain water molecules are also present here. Water molecules are also present in the case of cell membrane or plasma membrane. Now, before we go further, okay, let me tell you the inspiration. Yes, let me tell you the inspiration of your cell membrane. Okay, cell membrane is like an Indian road. Okay, wherever you're traveling, there will be a sudden hump or sudden pothole. Hump, pothole. It's going to keep happening. Same thing happens in the case of your cell membrane also. Cell membrane is said to be completely asymmetrical. It is said to be completely asymmetrical. It's going to be completely asymmetrical in nature. Okay. Now it is also said to be mosaic. It is also said to be mosaic. Now students, what is this mosaic, students? Can anyone tell me what is this mosaic nature? How many of you seen this? How many of you seen this in the houses? Like in the old houses, on the floor, this is the old fashioned granite. You might have seen this. This is called as mosaic. This is called as mosaic. Now, in this mosaic, can you see any pattern is available? Can you see any pattern here, students? Do you see any form of pattern here? Anyone, any pattern here? Any pattern? Absolutely not. It is completely random. Yes, it is completely random. So mosaic here means completely random. It's going to be completely random in nature. Mosaic like that. It is going to be completely random in nature. Now, plasma membrane is said to be living. It is said to be living, and it is also semi-solid. It's going to be completely semi-solid. This semi-solid has another name. It is also called as quasi-fluid. Quasi-fluid. What is this quasi-fluid now? Understand. Okay. If you see a typical cell membrane, right? If you take a typical cell membrane, it has something called as a solid. It looks solid, but it has a liquid property also. The meaning of what is liquid property? Now, all of you imagine and tell me, can you move around in a solid? For example, I have a wall here. Can you move from one side of the wall to other side of the wall? No. You cannot move through the solid. You don't have the ability to move through the solid. But what if there is a water? What if there is a liquid? Can you move through the liquid? Yes, you can. Like for example, water. You can go to swimming pool. You can nicely go swimming. You can. So here, the plasma membrane, even though it is solid, we call it semi-solid. Why? Because it also has a liquid property. That is, molecules can move around here. That is the meaning of quasi-fluid. The meaning of quasi-fluid is fluid-like movement can be seen here. Okay, fluid-like movement. Okay. Now, before we go any further, we need to understand what is the meaning of phospholipids. That is, what is the phospholipid bilayer here? What is the phospholipid bilayer? Okay, very simple. Just look at it. Okay, this is called as the round down head. Here we have tail, head and tail. This right here is a phospholipid bilayer. All of these are phospholipid. Example, lecithin is an example, right? All of these are lipids and they're present in a bilayer like this. And if you focus on one of them, right, this is how one of them looks like. Now, here, this part is called as a polar head. This is the polar head. And this right here is the non-polar tail. Non-polar tail. That is, this head right here, student, this right here is hydrophilic in nature. Hydrophilic in nature. And this tail here is going to be hydrophobic in nature. Hydrophobic in nature. Okay. This is the polar head and non-polar tail. Now I'll show you a better visualization. Okay, see this. Can you see? This is the entire head. That is the glycerol and this is the fatty acid. This is the hydrophilic head, hydrophobic tail. This is how they are arranged. Can you see? Entire cell membrane. They are arranged one top, all the heads in the corner, tails in the middle, tail in the middle. This is the diagram given in your NCERT also. But before we go any further, let me show you actually how it looks like. Okay. See, all of you see this. All of you see this. I hope the board is visible to all of you. Okay. Can you see here? I hope all of you are able to see. This entire thing is called as the polar head. Long polar head, non-polar tail on the middle. So polar molecules cannot pass through. Right? Usually polar molecules cannot pass through. Can you see here? This is going to be the entire. This is the entire plasma membrane. This is the layer. And this right here is how it looks like, the entire outer layer. Now here, can you see the different structures? These are called as the protein molecules here. Some protein molecules are present on the inside. Some protein molecules are present on the periphery. Can you see periphery? Now tell me, which one can you remove easily? One which is on the completely inside or one which is completely on the outside? Obviously, this can be removed out easily. Yes, that is called as periphery protein or peripheral protein. Okay, the protein which is present on the outside which can be easily removed, that is called as peripheral protein. This one. Okay, that is called as peripheral protein. Okay, where is it going now? Okay, it went. I think I lost it. Okay, students, that was the peripheral protein. Now, if you notice here also, in this NCI diagram, this one, can you see? One which is present on the outside here, this one. All of these are this one. One present on the outside. One which is present on the outside is called as peripheral protein. One which is present on the inside completely, it is called as integral protein. We also have cholesterol here, students. Cholesterol. Cholesterol helps in maintaining the stability. It helps in the stability of the entire cell membrane. Cholesterol. Okay. This is the cholesterol right here. Can you see, which helps in maintaining. This is the phosphobilipid layer. There is also sugar here, carbohydrates. There is also water here. Fluid mosaic model. Okay. This is the fluid mosaic model, students. Now, this fluid mosaic model was given by Singer and Nicholson. Singer and Nicholson. Singer and Nicholson in 1972. So, in 1972, Singer and Nicholson gave this entire structure. Okay. Now, here, remember I told you. Remember this, you can see here also. Lipid bilayer. Outer face, inner face. Integral proteins here. There are also certain channels here, different aquaporins here for the transport of water. Okay. Now, what is the movement here, students? Movement. I told you, it is quasi-fluid in nature. The meaning of quasi-fluid is that it can show motility, that there is a movement here. Okay. Now, there are two types of movements here. Okay. One is called as a flip-flop. Flip-flop movement. Other one is called as a lateral movement. Lateral movement. Now, what is flip-flop? What is lateral? You need to understand with the help of diagram again. Okay. Let's understand with the help of a diagram. Now, students, listen to me very carefully. Listen to me very, very carefully. All of you can see me, right? Am I audible? Sound check, sound check. Students, am I audible? Sound check, sound check. Sound check. Yes, I am audible, obviously. Okay. Now, see here. What is flip-flop? First, understand what is flip-flop right now. Flip-flop. Okay. If this one goes this side and this one goes this side, that is, from one phosphobilipid layer. This is moving this side and this is moving this side. That is called as flip-flop. You know that flip-flop? Is that a flip-flop? Okay. This is a flip-flop movement. One from here will go this side and this will go this side. That is called as flip-flop movement. And that can be shown by lipids alone. Lipids. Now here, in this entire lipid bilayer, there are proteins here. Proteins are here. Yes, proteins are here. Okay. Proteins are here. Now these proteins, right, these proteins will show movement. That is, this protein will move from here to here, That is, it will move like this, round, it will go. Okay, that is called as a lateral movement. Lateral. So it will only move in this plane, only one plane. Okay, so that is lateral movement. Lateral movement is shown mainly by the proteins, even by certain lipids also. Okay, that is the different types of movements: flip-flop and lateral movement. Clear to all of you? Clear all of you? What is flip-flop and what is lateral movement? Okay. Now what is the function? That is the most important, students. What is the function here? Okay. What are different functions? Can anyone tell me what do you think are the functions of cell membrane? What do you think is the function of cell membrane, students? Okay. Obviously, they are involved in the movement of proteins. They're involved in the movement of proteins. Their involvement is in the cell division, because during cell division the entire cell membrane will divide. Yes, it'll cut, it'll degrade, it'll open up and it'll split cell membrane. Some of cell membrane can also be involved in secretion. Okay, they can be involved in endocytosis. What is endocytosis? Basically, the entire cell membrane will undergo a vesicle formation. It will undergo a formation of vesicle formation like this. Can you see here? It undergo vesicle formation and it completely engulfs something. All of you might have heard a term called as phagocytosis. That is a type of endocytosis. Okay. Then there is also cell growth. In general, cell growth, and also there is intercellular junctions. Formation of intercellular junctions. So all of these are different functions of the cell membrane. Okay. Now let's understand a concept called transport. How does the cell membrane involvement in the transport? Now all of you might have heard about something called as active transport and passive transport. Some idea you might have. Initially this used to be an entire chapter. But right now one small basic you need to learn. Apart from that, nothing more. So all you need to learn is what is active transport and what is passive transport. That's all. Okay. Understand. First of all, understand what is passive transport. What is passive transport? Passive transport. What is passive transport, students? In the case of passive transport, no ATP is required. And this type of movement, right, happens along the concentration gradient. That is, it will always happen. From high concentration to low concentration. It doesn't require any ATP. It happens on its own. Now, one such example is by simple diffusion. Okay. By simple diffusion it can happen. Can anyone tell me what is that? This simple diffusion can be seen in the case of neutral transport. The neutral transport I'm talking about here is nothing but the osmosis. Osmosis can happen by simple diffusion. That is, water can move from high concentration to low concentration with ease. There is no ATP requirement, nothing is required, no energy whatsoever. Okay. But in the case of active transport, what is happening? That is, Here, usually the polar molecules, which cannot move across because of the non-polar tail and everything, polar molecules cannot move apart easily. So these polar molecules will attach to itself to a carrier protein, So these polar molecules will attach to a carrier protein, and with the use of ATP, they will travel from low concentration to high concentration. That is called as active transport. ATP is used. There is a carrier molecule here, Against the concentration gradient, low to high, very difficult. That is called as active transport. One famous example is sodium potassium pump. Sodium potassium pump is an example. Okay. Sodium potassium pump is an example here. Okay. Very simple. See, students. Now you might ask, students, is water polar molecule? Tell me. All of you tell me, students. Is water polar molecule, yes or no? Tell me in the chat quickly, all of you. Is water a polar molecule? Let me look at the chat. Tell me, is water a polar molecule? Tell me. All of you like the video? Target is 200 today. Target is 200 likes. So quickly smash like, keep smashing the like button. Okay. So if water is polar molecule, how does the water transport? See, I told you, water is polar molecule. If water is polar, it has to undergo active transport, no? How is it able to undergo passive transport? How? If water is polar, how does it travel? A question to all of you. Think. Use the brain cells. Use the simple brain cells. First, there are two main reasons. Okay. The first thing is the size. The size of the water molecules are so small. It is so small that it becomes negligible. For the transportation. That is the first thing, size. The second main point here is there are separate channels or canals, whatever you call it, called as aquaporins. Can you see here? There are certain separate canals called as aquaporins, and with the help of this aquaporins, water can transfer. So even though water is polar molecule, it can transport by simple diffusion with the help of aquaporins. All of you will remember that? All of you will remember, students? All of you will remember that? Yes or no? Tell me. All of you can remember that about aquaporins? This is called as aquaporins. Okay, aquaporins. Now we come to something called as cell wall. Okay. Now the first question to all of you, tell me, everyone, all of you tell me, cell wall is living or dead? Tell me, is cell wall living or dead? In the chat quickly. Is cell wall living or dead? Quickly in the chat. First of all, cell wall is non-living. Remember that point, all of you. It is completely non-living in nature. It is completely non-living. It is the outermost covering, all of you, yes or no? In the case of you, plant cell, it is the outermost covering. Yes, it is the outermost covering. It is found in the case of plant cell. Yes, it is found in the case of plant cell. And also in the case of certain protists. As well as fungi. It is found here. Okay. Now, students, before we go to the function, right? Before we go to the function of this entire cell wall, all of you know, main function is what? Protection. It helps prevent the cell from bursting, right? Provides the shape to the cell. All that they will never ask you. What they can ask you is, what is the cell wall made up of? See, if you look at a cell wall of a plant, this is a plant cell wall. If we look at the cell wall of a plant, it is made up of a layer of cellulose, hemicellulose, and pectin. So that they can ask you, what is cell wall made up of? And this is a NEET PYQ. Okay. So in the case of plants, what is it made up of, students? It is made up of hemicellulose, It is made up of cellulose. It is made up of pectins. Pectins. Now a question arises. What about in the case of even algae? Okay, here I forgot, right? Algae. See, algae also is plant. Algae always plant. But in the case of algae, the cell wall composition is a little different because algae are a little special because of the hydrocolloidal substance and also. So algae's cell wall is a little different compared to every single plant. In what way? Okay. For example, here also they have cellulose. Cellulose present here. But apart from cellulose here, there is something called as galactans. Galactans. There is something called as mannans. Mannans. There is something called as calcium carbonate. So all of these are present in the case of algae. Now, can anyone remind me, what about fungi? What about fungi? In the case of fungi, do they have something extra? Tell me, students. In the case of fungi, they have something called as chitin. Chitin can be found in the case of fungi. So please remember this. All of this needs to be in your book today only. Okay. Now I want every single student of mine today to make these notes. After today's class, there should be no backlog in this chapter because I have questions today. I have questions for all of you. Solve those questions. Be more confident. Okay. If you want all of these notes are available on the Telegram channel. Okay. Now, next concept. Okay. Now, what is this layer? What is this layer concept here in the cell wall? Because students, unlike your plasma membrane, which is lipid bilayer, cell wall has two separate layers actually. Some layer is present at a young stage. As the plant is growing older and older and older, there is a separate cell wall here. That is called as secondary cell wall. Now, how is the formation? What are the properties? Write down this much also. Okay, write down all of you. Now, for example, this is the primary cell wall. Let's do it in green, students. Green is better. Cell wall, no? Let's take this is the cell wall. Let's take this is the cell wall here. Okay. Now, students, this is called as the primary cell wall. Let's call it primary cell wall. Okay. Now, this primary cell wall which is present here, this primary cell wall is present in the young plants. In the young plants. This primary cell wall is also very permeable. It is also very permeable in nature. This primary cell wall is responsible for growth. Okay. Now what happens as the plant is growing older? As the plant is growing older, this primary cell wall will completely be diminished. It undergoes diminution. That is, on the inner surface, that on the from the inner side, can you see on the inside there will be formation of secondary cell wall. Can all of you see this? This right here is the secondary cell wall now. This right here is the secondary cell wall. And remember, students, secondary cell wall is always formed on the inside. It is always formed on the inside compared to the primary cell wall. Primary cell wall is outside. Secondary cell wall is on the inside. And this secondary cell wall, students, is completely non-elastic in nature. Non-elastic. It is non-permeable. It is non-permeable. Because non-elastic because it has certain depositions like suberin depositions, and also depositions like lignin depositions. That's why it is completely non-elastic in nature. Non-elastic and non-permeable. It is formed on the inside. Can you see inside? Now, how are the two cells attached to each other? That is a good concept here. That is, if you look at the two cells here, see, this is the primary cell wall. Inner side we have secondary cell wall. Okay. Now, for example, this is one cell, one more cell, one more cell. In between the cells, can you see? In between the cells, there is something called as middle lamella. I'll show you a better diagram. See here, students. This is one cell, this is one more cell. Outer part is cell wall. Between one cell and second cell, in the middle there is something called as middle lamella. And this middle lamella is rich in calcium pectate and magnesium pectate. It is rich in calcium pectate and magnesium pectate. Calcium pectate and magnesium pectate, middle lamella. Okay, that is middle lamella is present middle. Now here also, do you have middle lamella? We have middle lamella. Now, but students here understand, one cell and second cell, they can talk to each other. They can exchange material. How? There is a cytoplasmic connection here. There is a cytoplasmic connection. Cytoplasmic connection. Cytoplasmic connection. That is, cytoplasm here and cytoplasm here connected, and they are connected with the help of plasmodesmata. See, this is middle lamella here. In between the middle lamella, there is something called as plasmodesmata. Can you see here? Plasmodesmata. Plasmodesmata is a cytoplasmic connection which is connecting the two different cells in the middle. Is that point clear to all of you? Yes? All of you clear? Clear, students? That is plasmodesmata. Plasmodesmata right in the middle. Plasmodesmata right in the middle. Okay. Now, with that, cell membrane is also, cell wall is also over. Now, students, do you realize in the last, let's take around almost 2 hours, I believe. In the last 2 hours, you have finished around 70% of the chapter. No, not 70. 60% chapter is over now. 60% chapter is over right now. In the next 1.5 hour, it's a 3.5 hour chapter, right? In the next 1.5 hour, this entire chapter is going to be over, right? One by one, with the help of diagrams, I'm going to teach you the rest of the chapter. All you are left with right now, is endomembrane system. That is going to be your Golgi complex, endoplasmic reticulum, lysosomes and vacuoles. After that, you have mitochondria, chloroplast. Very simple diagrams, right? After that, I have to teach you something called as the most important part. That is going to be cilia and flagella structure and centriole structure. That will take some time, but we will finish it today only. One short lecture, one go. Okay? All of you. Can we start now? Can we start? All of you, drink some water. Okay? Drink some water. Now, students, I wanted to ask all of you that in your class 11th, right? So, no, plasmodesmata, right? Plasmodesmata is a connecting link between these two, right? It is a pipeline. Imagine there's a pipe going here, pipe. Middle lamella is like a glue. There. It is holding the entire thing. It's a glue there. Middle lamella is a glue here. Plasmodesmata is the connection here. Okay. Now, students, I wanted to ask all of you that is from your class 11th portion, what are your backlogs still left? Tell me honestly. I'll give you exact 30 seconds. I'll give you exactly 30 seconds to answer this question. Tell me from your class 11th portion. What are your backlogs left? Tell me. Backlogs. Let me see the chat. What is your backlogs from class 11th? Tell, it's fine. You can tell. I'm looking at the chat right now. And all of you quickly smash the like button. Right. Target is 200 today. All of you know the target? Yes. Photosynthesis. Recently I did no photosynthesis. Teju, go check out the channel. Last week I've done photosynthesis. Plant physiology I've done, right? Cell cycle I'll be doing next chapter. Students, all of you are watching the lecture right now. If you go to the channel right now, I have done 3D of both the plant physiology chapters. That is, respiration in plants is done in detail. photosynthesis in higher plants in detail. So promise me, students, promise me. All of you have that one chapter backlog, right? That one chapter as backlog. I want every single student of mine to finish that one chapter of plant physiology in this weekend. That is Saturday and Sunday. You have 2 days. You have 2 days time to finish that plant physiology backlogs. If you can finish, try finishing plant physiology, both the chapters, Saturday one chapter, Sunday one chapter. Promise all of you. Promise. Cell division, I will do it, don't worry. Cell division, I will do. Plant growth regulators, I will do later. Eventually, I will do plant growth regulators. It's very easy. Okay. Anatomy, morphology, I will do, don't worry. Can't solve PYQ. I'll make you solve, don't worry. So, in this Saturday, Sunday, you have 2 days, right? Teju, Tanuja, Chetu, all of you, right? In this 2 days time, Saturday and Sunday, try finishing all the backlogs. That 2 chapters, if you have 2 chapters, 1 chapter per day, you can do it easily, right? Botany, if it is there. Plant physiology, you can finish morphoanatomy, later we will do, don't worry again. Okay? All of you. Anatomy 3D is coming up, don't worry. Try doing photosynthesis in higher plants tomorrow. Sunday, you have to do respiration in plants. Okay, weekend plan. That is your weekend plan now. I have given you homework. I have given all of you some homework now. So all of you sincerely do that homework. Okay, sincerely do that homework. So please do the homework. Do not waste any time. Okay. So if you do these 2 chapters, you will realize your class 11th major backlogs is going to get over. Okay. Now, let's go to the next concept called as endomembrane system. Can we start, all of you? Can we start? Can we start now? All of you? Yes. Quickly some fire emoji in the chat. Or where is the bot army? Where is the bot army, students? Green hearts in the chat. Green hearts for bot army today. Okay. Now next concept is called as endomembrane system. Let me tell you what type of question will come here. I will tell you now only. The question we can expect here is the members, which are the organelles part of endomembrane system. That is the most famous question here. Famous question. Okay, very famous question. Okay, for that I have a small trick here. All my students, all of you know the trick, right? Every gamer loves victory. Everyone type it in the chat. Type. That is, every gamer loves victory. Every gamer loves victory. Okay. E for endoplasmic reticulum. G for Golgi complex or Golgi complex. L for gonna be your lysosomes. V for vacuoles. Okay, V for vacuoles. Now students, why are they part of endomembrane system? These organelles are part of endomembrane system. They are part of endomembrane system. All of them, students, all of them. They are part of endomembrane system because their functions are coordinated. Because their functions are coordinated. That is why they are part of endomembrane system. If you look here also, here we have Golgi complex, vacuoles here, lysosomes, and smooth and rough endoplasmic reticulum. All of them are part of endomembrane system because all of their functions are coordinated. Simple as such. That is the famous NEET PYQ. That is the famous NEET PYQ right there. Okay, famous NEET PYQ. Now the next question is going to be the endomembrane. That is going to be endoplasmic reticulum. Now here I want all of you to draw a diagram, simple diagram. Let's take a cell here. This is a cell right here. Let's say this is a cell. In this cell right here, this is the nucleus. Here we have the nucleus. Okay, nucleus. Now from this nucleus, students, from this nucleus has 2 separate layers, outer and inner. From this outer layer, we can see the development of endoplasmic reticulum. So endoplasmic reticulum will originate from the outer layer like this. Can you see? From the outer layer it will originate. So endoplasmic reticulum will always originate from the outer layer of the nucleus. Okay, nuclear envelope. Now here, all of you need to focus on small thing here. Now here, this is the part where we do not have the ER. Green color is the part where endoplasmic reticulum is missing. Part of cytoplasm basically. Now wherever we have red color, that is part of ER. That is the part of ER. That is the part of ER. Now this region, which is green color part, where endoplasmic reticulum is missing, this is called as extraluminal region. It is called as extraluminal region. Okay. Extraluminal region. Now what about this one, red color part? Red part is called as luminal region. Okay, luminal region. Okay. Now what did we understand from here? We understood that the entire endoplasmic reticulum can divide the entire cell into two parts, luminal and extraluminal part. Wherever we do not have ER, that becomes extraluminal. Wherever we have ER, that becomes luminal. So luminal, extraluminal, two different parts of your entire cell. Okay. But apart from all that, students, but what exactly is ER? Right? Endoplasmic reticulum is basically network, students. It is network. Can you see here? It is basically a network, right? It is a network or reticulum, or it is a network or reticulum of tubular structures. Reticulum of tubular structures here. Okay. Now we learned here it is an extension of nucleus. Extension of nucleus here. Now students, I want everyone to write down, this part is going to be the most important part here. Okay, that is, some of the ER has ribosomes on it. What do you call them? Such ER is called as RER. The meaning of R here is rough endoplasmic reticulum. Now some ER do not have that. That is no ribosome. Such an ER is called as SER. The meaning of SER is smooth endoplasmic reticulum. They are smooth because the ribosomes are missing there. Now both of these rough and smooth endoplasmic reticulum has a separate synthesis. For example, the rough endoplasmic reticulum will produce proteins, rough ER, while smooth ER will produce lipids and hormones, certain steroidal hormones. Okay, steroidal hormones. That is the SER. Is that point clear to all of you, students? Is this point clear to all of you? Okay, is this clear to all of you? Clear? Yeah. You can see here, extension of cell membrane, rough ER, smooth ER. Diagram given your NCERT, ribosomes are present on rough ER. Now, with that, we come to the next concept, right? Next concept is called as Golgi complex. Now, students, this Golgi complex was actually, it is named after a scientist who discovered it. The scientist who discovered it is called as 𝐂𝐤 Golgi. His name is 𝐂𝐤. So what name? I don't even know what name they keep. Our names are so much better, no? So easy names we have. 𝐂𝐤 Golgi. I don't even know. I can't even comprehend if someone name is 𝐂𝐤. 𝐂𝐤 Golgi. Okay. So 𝐂𝐤 Golgi in the year 1898, almost 100 years before I was born. No? Nice. 1898, okay. He found certain densely stained structures, densely stained structures, densely stained structure, okay, near nucleus, near nucleus, and he called it as Golgi body, Golgi body. But here, students, unlike unlike the endoplasmic reticulum, Golgi bodies are much more flat. They're much more flat. They're much more parallel to each other. I'll show you. I'll show you. It'll be easy to visualize it. Can you see here? This is 𝐂𝐤 Golgi. Okay, nice mustache. Can you see? They're much more parallel to each other. They're much more parallel to each other, and they're concentrically stacked here. Yes, they're concentrically stacked. Yes. Okay. They're going to be flat and disc shaped. Flat and disc shaped. They're going to be flat and complete disc shaped, or we can say disc shaped sacs. Disc shaped sacs. Okay. Here, did you see this? This part, this one, this long structure. They're called as cisternae. They're called as. Now, students, how many of you find it difficult to remember cisternae and cristae? Cristae is part of mitochondria. Cisternae is part of Golgi complex. How many of you get confused between these two? Here is a simple solution. Tell me which one is longer to say, cisternae or cristae? Which one is longer? Cisternae, right? Cisternae is longer to say? Longer to say. Golgi complex is longer again. So Golgi complex also is longer to say, Golgi and complex. So longer term, longer. So that's how you can small correlation you can do. Okay. So cisternae and Golgi complex, cristae, mitochondria. Please don't get confused between these two. I'm very sure many will get confused between these two. Okay. That is cisternae. Cisternae are concentrically arranged. Can you see concentrically arranged? Now here, there's a famous concept here. There's a famous concept here. Understand? The famous concept here is the face. How is the face here? For example, this is the nucleus. From this nucleus, do we have certain structure like this? What do you call that? Let's just say this is nucleus. This is nucleus. And this right here is the ER. Okay, this right is the ER. Okay. Now, near to ER, we can find the Golgi complex here. Okay. Let's take, we have Golgi complex here. Okay. We have Golgi complex here. Now, this Golgi complex, which is near to ER, this Golgi complex, okay, let me write for you very carefully. Listen to me very carefully. This Golgi complex has two different faces. Two different faces. In front of you there's something, behind your back there's something, right? People have two faces. Similarly, a Golgi complex also has two separate faces. What are the two different faces? Understand. Here, can you see this part which is facing towards the nucleus? Can you see this part? This part is called as cis face. Here, where do I write here? Right, let's write here. This one is called as cis face. Cis face, or also called as convex face. Convex or forming face. Cis face, convex face or forming face. One which is near the nucleus, okay, cis face. Now, one which is going away from the nucleus, this side, away from the nucleus, this side, away side. This part is called as trans face. Trans or concave or maturing face. Now, is this important to learn? Which face is this side, which face is that side? It is important to learn. Very important. Why? Because the function is very important to learn here. The main function of Golgi complex, is to do packaging. Nicely package. Now, how does it do packaging? For example, from this ER, certain things are released. Let's say a protein is released or a lipid is released. Now, this lipid or protein will first enter or first reach the cis face, convex face or forming face. It will reach here. Now from here, it undergoes packaging, and becomes exit here. It will exit here in the form of vesicles. In the form of vesicles, it will exit here. One more time I'll explain. Listen to me. Here, can you see here? Here we have cis phase. Cis phase, also called as forming phase or convex. Here we have trans phase. This side is trans phase. Trans phase is also called as concave, or it is called as maturing phase. Now here, every single lipid or a protein, it will come from where? ER. It will come from here. It will encounter this phase first. Then it undergoes packaging, and exit from here. I'll show you one more. Diagram will make it easy for you to understand. See here. First, here we have what? Nucleus. Near the nucleus, what do we have? ER, endoplasmic reticulum. Now this endoplasmic reticulum will produce protein. It can produce lipid from here. It can produce lipid. This phase, students, tell me, what is this phase called as? Where it is first receiving it? Which phase is receiving the proteins, or which phase is receiving the lipid? This is called as the cis phase. On the cis phase, it is entering. Now it undergoes packaging, and finally it is released in the form of vesicles. In the form of vesicle, it is removed from the trans phase. From trans phase, it is coming out. So cis entry, trans is exit. Is that clear to all of you, students? Is that clear to all of you? Simple concept. Is that clear to all of you? How does the packaging happen? Cis phase is entry, trans phase is exit. Cis and trans. Cis phase is entry, trans phase exit. Now I will make you understand the function. You will get the proper idea right now. The bigger picture you will understand. Okay. Now what is it produced? It can produce two main things. One is called as glycoprotein, other one is called as glycolipid. Okay, understand, very simple it is. Okay. Glycoprotein, and it will produce glycolipid. Okay. These are the products of your coming from Golgi complex. Okay. Now here, this protein is coming from RER. This lipid is coming from SCR. Now, what is the main function of Golgi complex? The main function of Golgi complex is to add glyco part. That is basically a type of glucose or carbohydrate is added. Understood? So glycoprotein and glycolipids are produced by Golgi complex. This glyco is added to protein and sent out. Add lipid and sent out. That is all is the function of Golgi complex. That is packaging. Nice little package. Amazon factory. This is Amazon factory. Okay. Amazon factory, students. Dictyosomes are the Golgi complex in the case of plant. We call plant Golgi complex as dictyosomes. Plant complex is called as dictyosomes. Okay, Meghna? Dictyosomes. Now, students. How many of you heard about suicidal bags? Very easy concept. Okay. Very easy concept. Here it forms something called as vesicles. It forms vesicles here. That is, lysosomes are formed by the budding of vesicles from the Golgi complex. See here. These are the lysosomes. Can you see? Suicidal bags. These are the suicidal bags here under microscope. Now here do we have Golgi apparatus? Golgi body. From this Golgi body, can you see it, students? This is the cis phase, this is the trans phase. From the trans phase, we are getting the lysosomes are coming out. Lysosomes are coming out. If you see here also, nucleus, endoplasmic reticulum, Golgi complex, cis phase is entering, trans phase it is coming out, and that is producing the lysosomes. That is producing the lysosomes. So budding of vesicles from the Golgi complex is producing the lysosomes. It is producing the lysosomes. Now students, these lysosomes, right? These lysosomes has something called as lysosomal vesicle, right? This is a lysosomal vesicle. These lysosomal vesicle has something called as hydrolytic enzymes. Lipases, carboxylases, right? Proteases, carbohydrates. It also has something called as lipases, and there is other different enzymes, basically different types of hydrolytic enzymes, lipases. But for all of this different types of enzymes to function, there is a requirement. For all of these enzymes to function, there is a requirement. The requirement is acidic pH. It requires acidic pH. If there is no acidic pH, all of these enzymes will become deactivated. All of you know, in biomolecules we learned that every single enzyme has an optimum pH or optimum temperature at which it will function. So if the pH is not acidic enough, all of these enzymes will become denatured or inactivated. Clear, all of you? Clear students? Acidic pH is important. Without acidic pH, they cannot survive. Okay. Now, what is vacuole? Vacuole is like a dustbin. Vacuole is like a dustbin. All of this excretory waste, and all of these different types of plant sap, mainly in the case of plant, right? All of these plant sap, everything is stored inside the vacuole. It is basically a dustbin here, students. Dustbin. It is, first of all, it is a membrane bound structure. It is, obviously it is membrane bound because it's part of endomembrane system. Inside this vacuole there will be water sap. All the water sap is stored here nicely, as also excretory waste is stored here. Now students, here there's one small thing here which you need to understand here is, here in the case of plant cell, 90%, right? 90% is the cell volume. That is, in the case of plant cell, vacuoles becomes 90% of cell. See, can you see? In this plant cell, almost 90% is only vacuole. That large vacuole can be present in the case of plant cell because it has a lot of plant sap to store. Okay. Now here, if you notice, this entire vacuole has a single membrane. I told you here, membrane bound. It has a single membrane, and the name of this membrane is called as a tonoplast. It is called as a tonoplast. Can you see here? This is called as a tonoplast. Outer layer is called as tonoplast. Now there is a special function here. What is the function? This tonoplast right here, this outer layer is called as tonoplast. Now this tonoplast will make sure that there is a transport of ions inside. There will be lot of ions on the inside of this tonoplast, even against the concentration gradient. Okay, even if there is low ions here, low ions will go inside. That is the tonoplast layer, outer layer called as tonoplast in the vacuole. Okay, vacuole. Now, are there any different type of vacuoles? Yes, there is. In the case of plants, there is something called as sap vacuole. It is there. In the case of certain protists, there is something called as amoeba and paramecium. In the case of amoeba and paramecium, they have a different type of vacuole. Okay. For example, in the case of amoeba, there is something called as contractile vacuole, and this contractile vacuole can help in osmoregulation. It can help in osmoregulation. Okay, regulation of all the different salts also, and it can also help in excretion. That is contractile vacuole in the case of amoeba. But in the case of certain protists like paramecium, they have something called as food vacuole. Can you see food vacuole? This food vacuole will help in ingestion of food. That is food vacuole. Okay. So contractile vacuole, food vacuole. So students, that is the complete end of your endomembrane system. Golgi complex, endoplasmic reticulum, lysosomes and vacuoles. Every main point I have mentioned to you, and I want every single student after this class try reading NCERT just once. This entire chapter is going to become such an easy job for you, such a cakewalk. Okay, such an easy job will become. Yes. So students, how many of you are making notes? Raise your hands. How many of you are making notes? Raise your hands. How many of you are making notes? Raise your hands. On the spot. Spot notes. How many of you are making spot notes? Or how many of you are waiting for the PDF? How many of you are waiting for the PDF? How many of you are making the notes? Raise your hands. Raise your hands quickly. Raise your hands. How many of you are making the spot notes? How many of you are making the directly you're studying? Quickly. Already done. Nice. If you're already done, it's even more amazing. Okay. So students, try to make the spot notes, because let me tell you, right, if there is a, see, I'm not also perfect. I am not always perfect, right? If there is any missed out point, I would recommend all of you to open your NCERT. I would recommend all of you to open your NCERT and read the lines. Just try reading the lines of NCERT. It's my guarantee from my side to you. If any point is missing, add to these notes. Add to these notes and it will benefit you. All of you will do that? Okay. Yes, Christina, acrosome. I didn't tell you that part. Okay. One small information you want me to tell, I'll tell you. Okay. In Golgi complex, where can we find Golgi complex? Golgi complex can be found in the acrosome. All of you know what is acrosome? The cap of the sperm, right? And also in the root cap. Also in the root cap we can find Golgi complex. But in the case of root cap, the Golgi complex will make sure the root cap is much more mucus in nature, right? If it is mucus in nature, the root cap can easily penetrate in the soil. That is not given in your NCERT, that's why I didn't tell you. Okay, today I'm trying to keep it as much as closely associated with NCERT today. Okay, done? What? Mr. Perfect, it seems. joke you're telling, joke. Okay, now students, can we start mitochondria now? Why? Because mitochondria is called as what? Mitochondria is called as what, students? Can anyone tell me what is the another name of mitochondria? Everyone in the chat tell me. What is the other name of mitochondria? All of you know this, right? All of you know this. The other name of mitochondria, it is called as powerhouse of the cell. Powerhouse of the cell. Powerhouse of the cell. Now the question to all of you, why? Why do we call it as powerhouse of the cell? It is called as powerhouse of the cell because it can produce ATP. Because it can produce ATP, adenosine triphosphate. Adenosine triphosphate. How? Because of oxidative phosphorylation. From oxidative phosphorylation in ETC, electron transport chain. Electron transport chain, it can make ATP. It can make ATP. Okay. Now students, I'll ask you one small thing. What does it look like? Doesn't it look like a sausage? It does. So we call it a sausage shape. We call it as sausage shape. Okay. It is the site for aerobic respiration. Site for aerobic respiration. All type of aerobic respiration occurs here. Aerobic respiration occurs here. And this structure is single or double? It is double membrane. Double membrane structure. It is a complete double membrane structure. Clear, students? Clear, all of you? Clear? Is visible? See here, students. This entire thing is called as a mitochondria. Now can you see here? It has a outer layer. It has a outer layer here. Outer layer. And it has a inner layer. It has a inner layer like this. This inner layer is called as cristae. This inner layer is called as cristae. The fascinating thing about your entire mitochondria is that this is a structure which has its own DNA. It has its own ribosomes. And the ribosome present here is actually 70s ribosome. Okay, can you see here, all of you? Ribosome present here. This is the structure of mitochondria. This is the structure of mitochondria right here. Okay, see here. This is the mitochondria. It is completely double membrane structure. Double membrane structure. This is the NCI diagram. This entire crista is the enfoldings. Matrix is going to be the entire cytoplasm-like structure. There is intermembrane space. Now students, I want you to draw this. Let's draw this. It's very easy to draw, right? Very easy to draw. I'll give you a top view. Top view of this entire mitochondria. Okay. Top view of this mitochondria. Write down, draw this. Now here, what do we have, students? Here we have the outer membrane, and we have the inner membrane. Inner membrane. Outer membrane, inner membrane. Between the two membrane, intermembrane space. We also have intermembrane space. Intermembrane space also is available here. Done? Done. Now students, here the folding from the inner membrane itself, there are foldings here. Can you see? These are the different foldings. These foldings are called as cristae, right? Which color should I write? Okay. What do they do? These cristae help in increasing the surface area. Increasing the surface area. Okay. Now here I told you there is own DNA is present here. Yes, there's a DNA present here. Circular DNA. Okay, this DNA is here. This DNA here is double stranded DNA. Double stranded DNA. Okay, double stranded DNA. But this DNA does not have any histone protein. It does not have any histone protein. Double stranded DNA. Apart from that, there is also ribosomes here. These ribosomes are just like prokaryotic ribosomes. They are 70s ribosome. They are 70s ribosome. Okay. Now, students, apart from all that, there is a main structure which is present here, which helps in which mainly helps in the entire process of your ATP synthesis, is called as F0 F1 particle. It is also called as oxysomes. It is also called as ATP synthase. Can you see here? There are certain round such like this everywhere. Okay, these are the structures here. These are the structures here. They are called as elementary particle, or they can be called as oxysomes, They can be called as oxysomes, or they can be called as ATP synthase. ATP synthase. We can also call them F0, F1 particle also. All different names. F0, F1 particle. Okay, this one. How does it look like? Here. This is how it looks like. Can you see here? Everywhere it is going to be present F0, F1 particle. See here? F0, F1 particle. F0, F1 particle. Okay. So that is the end of one double membrane structure called as the mitochondria. Now the next one is called as double membrane structure called as plastid. Let me tell you, this plastid, right? Here under plastid we have a structure called as chloroplast, and this plastid right here, it is present only in the case of plant and completely absent in the case of animal cell. Completely absent. Okay, this is the entire plastid. Okay, entire plastid. It is present in all plants and certain euglenoids. Okay, now here what happens is a plastid can impart a specific pigment, and the specific pigment can produce a certain color. For example, there is something called as chloroplast. Okay, now this chloroplast can help in what? Students, tell me in the chat quickly. This chloroplast can help in photosynthesis, and it imparts green color. That is chloroplast. Now there are two types of chloroplast which can mainly help in photosynthesis. That is basically chlorophyll, right? Chlorophyll as well as certain carotenoids. That is the chloroplast here. Green color they will give you. Okay, they help in photosynthesis, or I can say trapping light. All of you remember the main function of this trapping light is basically they are part of something called as students what? LHC, light harvesting complex. All of you remember chlorophyll A. Chlorophyll A is the reaction center. Apart from chlorophyll A, there is also certain small antennas, that is chlorophyll B, xanthophylls and carotenoids. So the main function is trapping the light by becoming part of LHC. Okay, LHC. Now, second one is called as chromoplast. Chromoplast is the first one. Second one is called as chromoplast. Now, unlike chloroplast, chromoplast does not help in photosynthesis, but chromoplast will impart color. Which type of color? It will impart yellow, orange, all that color. You see, right? Leaf. Different leaves, different fruits. Yellow color in the fruits, red color in the fruits, orange color in the fruits. All that is because of chromoplast. The examples here are carotenoids again. Carotenoids and xanthophyll. Xanthophylls. That is carotenoids, chromoplast. The third one is called as leucoplast. Now students, tell me, do you think this leucoplast, do you think it provides any color in nature? Does it provide any type of color in general? No. It does not provide any color, but this leucoplast main function is storage. It can help in storage of different substances. For example, under leucoplast, there are three main categories under leucoplast. Okay, the first one is called as a myeloplast. Second one is called as your ileoplast. And the third one is called as ailuroplast. Now students, what does it help in storage? Okay, For example, amylopectin. Amylopectin are part of starch. Yes or no? Tell me. They help in starch storage, or we can just say carbohydrate storage. Help in carbohydrate storage. Now, eleoplast, oil and fats. Aileron layer. Do you remember in the case of seed we learned? Aileron layer is a proteinaceous layer. Aileroplast helps in storage of protein. It helps in storage of proteins. So that is the three different types of plastid. Now here we have a diagram of your entire chloroplast also. All of you know chloroplast is a double membrane. So outer membrane, inner membrane. Now here, if you know the chloroplast diagram, like chloroplast, it has something called as stack structures. They are called as thylakoid. Each individual is called as a thylakoid. This entire stack is called as grana. Entire stack is called grana. And this grana, they are connected to each other with the help of stromal lamella. Stromal lamella is a connecting link between two different granas. Okay? Two different granas. Let me show you, students. You'll get a better understanding. See here, students. This entire thing is the chloroplast. This entire thing is called as a chloroplast. Can you see here? It has outer membrane and inner membrane. Outer membrane, inner membrane. Now, similar to mitochondria, even the chloroplast has its own DNA and own ribosome. The ribosome present here is 70S ribosome again, similar to the entire mitochondria. Now, if you look at here, these are called as the thylakoids. Can you see here? This entire stacks is called as thylakoid. One coin is called as thylakoid, but this entire stack is called as grana. Now this grana and this grana are connected. Can you see connected? The link between them, that is called as lamellae, stromal lamellae. There is DNA here, there is lipids here, there is ribosomes here are available because there is one DNA, right? That is the structure of entire chloroplast, students. Entire chloroplast given here also, chloroplast. Okay, now students, we come to ribosomes again. Now this is the electron microscope diagram. Students, this right here is the electron microscope diagram of your ribosome. Now one question arises, I think it was a very old question. Who discovered ribosomes? Can anyone tell me in the chat? Anyone? Who discovered ribosomes? Quickly in the chat. Students. Who discovered ribosomes? Anyone? I'm looking at the chat. Let's see who can tell the answer. George Paldin, exactly. George Paldin. Okay, George Paldin in the year 1953. Okay, so George Paldin in the year 1953 discovered the ribosomes. Okay, he found certain densely chained particles and he called it ribosomes. Now here I want you to understand two main things. First of all, what is ribosomes made up of, and what are different types of ribosome here? Okay, listen to me very carefully. What is ribosome made up of? Ribosomes are made up of RNA and protein. First point clear? Ribosomes are made up of RNA and protein together. RNA and protein. That is amino acid. Types of ribosomes. In the case of prokaryote, we have 70s ribosome. In the case of eukaryote, we have 80s ribosome. Here 80s is divided into 40 and 60. 40 and 60. This is your 80s ribosome in the case of eukaryotes. And that's all. Nothing more than this they'll ask you. Nothing more than this they will ask you. So ribosomes, very simple concept. Who discovered? What is ribosomes made up of? What does it help in? It helps in protein synthesis. Yes. And what are different types? 70s in the case of prokaryotes, 80s in the case of eukaryotes. Okay. Done, students? All of you? Now, students, let's take a 5 minutes break. Right. Let's take a 10 minutes break, students. Let's take a 10 minutes break. I will drink some water and come. Right. Let's take a 10 minutes break and then we will start. Okay? Or let's take a 15 minutes break itself. Let's take a 15 minutes break. I will go. I will drink some water and come. I'll sit for some time. Exactly at 9 o'clock, we'll start again. Okay? No. No. Ribosomes mainly has the RNA. Right? It mainly has the RNA here. RNA. Okay. So let's have a break now. What's the time right now, students? 8:45, right? 8:45. Okay. So I'll see you all of you at exactly. Don't worry, I have a lot of quiz. I have a lot of questions later on. We'll do the questions later on. Okay. So we'll take a break till 9 and we'll start again. After that, we have around, like, let's say around 45 minutes class, that's all, or 30 minutes class. Sound check, sound check. Yes. Now students, welcome back. Okay. Now, listen to me very carefully, right? Your body has a lot of backbone to it. When I say backbone, I mean you have a lot of bones in your body, right? You have different types of bones in your hand, different type of bones in your leg, different type of bones in your brain. That is not brain, the head. The skull is there, right? So your entire body has a framework. Those are the bones. Similarly, a cell also has a backbone or the support. The support or the backbone of a cell is called as cytoskeleton. So cytoskeleton, right? Cytoskeleton. One second, students. Yeah, sound is coming. Both the length and diameter of mitochondria they usually don't ask, but I think I wrote it in a diagram there. I believe length and diameter of a mitochondria was mentioned here somewhere. Don't worry, it's already. I think it's mentioned it somewhere here. Here, this is the length. This is the diameter. Length, diameter. They usually don't ask that. That's why I ignored it. Okay. Now, students, cytoskeleton you understand. Very simple. It is. It is basically the network of protein structures. It is a network of protein structure which provides the entire framework, provides the back framework. Now students, if you read this in your NCERT, you probably will not understand. I wanted to write down the few things which I'm going to teach you right now because this is what is important to understand the remaining topics. If you want to understand what is the structure of cilia and flagella, what is the structure of the entire centrioles, this point of time, that is the cytoskeleton, is very important to understand. Listen to me very carefully, all of you. Okay, there are in total three different types of cytoskeleton. Okay, three different types of cytoskeleton. The first one is called as microtubules. Microtubules. Second one is called as microfilament. It's called as microfilament. Okay. Third one is called as intermediate filament. Now, what is the difference between all of them? Understand. Very simple. It is. Follow. This one is said to be hollow completely. It is completely hollow in nature. Microfilaments are comparatively solid, and intermediate filaments are also hollow in nature. I will show you a diagram. You will understand better first of all. Okay. Now, all of you follow. This is unbranched, and this also is unbranched. Unbranched in nature. Unbranched. This is non-contractile. It's going to be completely non-contractile. Macrofilaments are comparatively contractile. They can basically contract like myelin. All of you know actin and myelin. Actin filaments, they can contract. This is contractile. Okay, contractile. This is again non-contractile. Non-contractile in nature. Non-contractile. Now students, write down the most important part. Where do you find microtubules? Microtubules can be found in cilia. It can be found in flagella. It can be found in the case of centrioles. So all of these major organelles, right? Cilia, flagella and centrioles, they are made up of microtubules. So please remember that. Because when I show you the diagram of microfilaments or the diagram of your cilia, flagella and centrioles, you need to tell me, sir, it is made up of microtubules. It is made up of microtubules. Clear? Clear all of you? Okay. Now students, understand cilia and flagella right now. Okay, cilia and flagella, and where do you find them, and what is the difference between cilia and flagella? Okay, what is the main difference between cilia and flagella also? Now I'll show you a diagram. See this. Where is that? Where is that? Now, for example, this is a eukaryotic organism. Can you see here on the outer surface this round line-like structure here out? Can you see here? This right here is called as cilia. This is called as cilia right here. Cilia. And when this cilia moves, if the cilia is moving, this entire organism can move around. Here is cilia, right? Cilia. Same cilia is also present in the fallopian tube also. Cilia. Ciliated movement. Okay. Now, can you see this is a flagella? Now here, this is the cilia here, and this is the flagella here. Long flagella. Okay. So basically, all the cilia and flagella are hair-like structures. They're basically hair-like outgrowth. Hair-like outgrowth. Hair-like outgrowth of cell membrane. They're coming from cell membrane directly. Of cell membrane. Okay. Now, where do they arise from? They emerge from basal body. They're coming out of basal body. Now, what is basal body and how does it look like? That is very important because this is a line in NCERT, doesn't give you better clarity. See this. Students, can you see here? This entire part is called as basal body. This middle down part is called as basal body. From this basal body upwards is the cilia and flagella coming out. And as you can see, this cilia and flagella are tube-like structure. Can you see? They're completely unbranched. They're unbranched, non-contractile. They're just assembly here and they're completely hollow. Can you see from top view? They're completely hollow. So all of this structure, this is the structure of cilia and flagella from the inside. So how does the cilia and flagella look from the inside? This is the thing. Can you see? Cilia and flagella from the top view. Okay. This is called as basal body, and this is the entire cilia and flagella. Okay. Now understand, students. Difference you understand, cilia and flagella. Okay. Small difference is there. Cilia and flagella. Okay. Now cilia is much comparatively shorter. It is going to be shorter, or we just say smaller, while the flagella is going to be comparatively longer. It is going to be comparatively longer, flagella. Now cilia, students, cilia shows oar-like movement. Now what is this oar movement, students? The meaning of oar is this. This is oars. In a boat there is oars. Now if I hold the oars and if I move it around, the oar, will the boat move front and back? It will move. Depending upon how you are moving it, you either will go front or back. Similarly, this cilia will move like that. It will push the fluid around it. It will push the aqueous medium around it and it will go forward. Cilia will help in movement. Cilia will help in movement here. Cilia will help in movement. Okay. Cilia will help movement. Now cilia is present throughout the body. Throughout the body we can find cilia. But flagella is generally present at the end. It is generally present at the end. Now, what will they ask you question here? The main question which they ask you is, what is the structure of cilia and flagella? That is the arrangement of microtubules. How are the different types of microtubules arranged in cilia and flagella? That is the main question they can ask you. See the diagram, all of you. Now, this is cilia, this is flagella, right? Now, this is the diagram given in your NCERT. Let's draw this entire diagram in a much more easy manner now. Okay. Same thing is given here also. Can you see? All of these are the microtubules. Microtubules. Now, I'll make you draw one more. That is my diagram. Ready, all of you? Ready? Now, I want everyone to draw this. I want everyone to draw this in your book. Let's the structure of cilia and flagella. This is the entire outer area. Okay. Now here in the middle we have two microtubules in the center. Let's just say in the center we have two microtubules in the center. two microtubules in the center. Okay. Now these two microtubules are joined together by a bridge here. Bridge. Now this is the central microtubules. Around the central microtubules, there is a central sheath like this. There's a central sheath like this. Okay. Now from this central sheath, there will be some structures coming out. 2, 3, 4, 5, 6, 7. One second. 5, 6, 7, 8, 5, 6, 7, 8, 9. 9 spikes are coming out. Okay. Now at the end of these spikes, there is also more microtubules. 2, 3, 4, 5, 6, 7, 8, 9. 9 microtubules are present on the surrounding also. Okay. Now, all of these are also attached to each other. BA. There's a BA linker here. Now these are attached to each other. These individual ones are attached to each other. Can you see? They are attached to each other. Now what is this entire structure? Let's do labeling here. This entire structure here, students, this entire structure is called as core. This entire structure is called as core. Can you hear me now? Okay, calm down, students. Am I audible now? Am I audible to all of you? Oops, suddenly when this thing happened. Am I audible? Sound check. Don't worry, I'll give you the entire Entire thing I'll give you, don't worry. One more time revision I'll give you, don't worry. Yes, right. Not audible. Still not audible. Okay. Listen to me all of you very carefully. Listen to me all of you very carefully now. What does this diagram represent? Okay. I think I'm audible right now to all of you. How long will it take to see the chats? I was in the zone. You know, when you're in the zone, you're in the zone. Okay. Now students, listen to me. This is the structure which we are drawing right now. Can you see? This is the structure which we are drawing right now. This is the basal body. This is the long flagella of cilia. This is top part. This is top part here. This top portion we are drawing. We are drawing the top portion from here. Okay. This is what is given in your NCERT. This is the electron microscope here. This is what is given in your NCERT. This is what we have drawn. Okay. Now here, do you see this? This entire thing is called as the core. Right. This entire thing is called as core or axoneme. Right. Core or axoneme. Now students, in this core or axoneme, here we have peripheral microtubules. Peripheral microtubules. Can any one of you count and tell me how many are there? How many do you think there are peripheral microtubules? Just count. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are totally 9 peripheral microtubules. 9 are there? 9 are there. Now there are 2 central microtubules. 9 plus 2 central microtubules are there. Data is there. Okay. Now this pink color part is called as what? Central sheath. Is called as central sheath. Now this central sheath is connected to the periphery. Central sheath is connected to periphery by radial spoke. Now between the two periphery microtubules, we have a linker. That is Ba linker is present here. Ba linker is present. And this is connected by a bridge. This is connected by a bridge here. And this is the entire structure of cilia and flagella, which shows 9 plus 2 arrangement. 9 periphery, 2 central. So 9 plus 2 arrangement of cilia and flagella, the structure. Done. That's all. They might ask you this entire structure, so please be careful. 2 central, 9 periphery. Okay. This is how it looks. Can you see? This is how it looks. This is how it's actually looking. So you need to know what you're seeing. You are seeing the top view of a cross section. You are seeing the top view of a cross section of a cilia and flagella, which is microtubules, hollow. Okay. Now students, understand centrosome. Understand the centrosome. Centrosome is completely non-membrane bound. Very important to understand. It is completely non-membrane. It is completely non-membrane. Remember, I told you in the starting only. Centrioles as well as ribosomes, both of them do not have any membrane. They're membraneless. So they do not have any membrane here. Non-membrane bound. Now the question to all of you, if you remember cell cycle chapter, centrosomes, do they duplicate? Yes, they duplicate. They duplicate in the S phase of cell cycle. S phase of cell cycle in the cytoplasm. S phase of cell cycle, they duplicate. Okay, they're non-membrane bound. They duplicate in S phase cycle. Now students, what is a centrosome and what is a centriole? Understand the difference between centrosome and centriole. Minor difference, but very important. Okay. Now understand. I'll draw a small diagram here, I'll draw a bigger diagram there. Okay. Here, this entire structure is called as centrosome. This entire structure is called a centrosome. Do you see this small perpendicular structure, 2 sticks like structure? These are called as centrioles. They are called as centrioles. So this entire thing is called as a centrosome. These 2 things are called as centrioles. These centrioles are said to be in 90 degree to each other. 90 degree to each other, that is perpendicular to each other. Centrioles are perpendicular to each other. Can you see here also? Entire thing is called as a centrosome. This is called as perpendicular to each other. That is called as centrioles. Okay. Now students, this entire structure has a amorphous coating on the outside. Can you see? that is called as this entire something membrane like. It is not membrane actually, it is a outer covering, that is called as amorphous pericentriolar space. Write it here down. That is called as amorphous pericentriolar. Okay, I mean, it can also be called as kinoplasm. Some textbooks will say kinoplasm. Some textbooks will even call it as centroplasm or centrosphere. Centrosphere. Some of them will call it centrosphere. Same meaning, this outer part. Now students, now we will learn how this particular structure, this centriole looks from a top view. From top view, how it looks like, because it is also made up of microtubule. It means that this also is very hollow in shape. Yes or no? Tell me. This also is very hollow. One second, students. Let me show you an image. This is very important to understand. Now, can all of you see this? Students? This is a centriole. This is centrioles. Two different centrioles. Are they perpendicular to each other? All of you can see they are perpendicular to each other. Now, students, these centrioles, two centrioles, are also made up of microtubules. They are made up of microtubules. Now, if you look at these microtubules from top view, do they look hollow? Can all of you tell me, are they looking hollow? Yes, they're looking hollow. Now here also, all of these microtubules are arranged in a fashion. They're arranged in a fashion. Okay. Now this is what we're going to learn, and this is what we are going to draw now. So all of you need to get that clarity in your head, what exactly we're trying to study here. Okay. So centrioles from the top view, how do they look like? Okay. This is also called as cartwheel shape. Okay. I hope all of you are able to follow this. This is called as centriole. Okay. Now, students, wait a minute. Let me show if I can find a image for your cilia and flagella. Also, you can get a better idea. Also. Okay. Give me a minute, students. Give me a minute. No, got it. Students, did we just learn this? Students? This is called as the basal body. Did we learn that? This is called as the basal body. Entire thing is covered by a plasma membrane. And here we have the top view. Can all of you see the top view? This is the entire top view they have given here. Top view. Now, did we just learn that there are totally how many peripheral? 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 periphery microtubules. 9 are there. Are they in doublet? They are in doublet. Okay. Can you see doublet? This is called a central microtubule. This is called a central sheath. These are called as radial spokes. They are called as radial spokes. So this was cilia and flagella. Students. This is cilia and flagella. Okay. This is called as cilia and flagella. So I hope all of you are able to visualize and understand what exactly cilia and flagella is. How does it look like? Okay, cilia and flagella. Okay. Now, this cilia and flagella. Now, that is over. Now we are understanding what is centrosome. Now, how does the centrosome look from the top view? Do you remember this? Students, do you remember this? How it looks from the top view? Now, let's draw this. All of you. Let's draw this. Okay. We get a better understanding if you draw, right? Right? Now, unlike your cilia and flagella, in the case of centrioles, in the middle there is no microtubule. Okay. Now here we have in center we have a hub, proteinaceous hub. Now from this proteinaceous hub there are 9 radial spokes going outside. 1, 2, 3, 4, 5, 6, 7, 8, 9. There are totally 9 radial spokes are going outside from here. 9 radial spokes. Now at the end of 9 radial spokes, there are peripheral microtubules. Peripheral microtubules, but these peripheral microtubules are present in triplets. Can you see, students? They are present in triplets here. Okay, so these are the peripheral microtubules. They are the peripheral microtubules. This right here is the radial spoke. Radial spoke. Now here in the middle, we have a structure called as hub. This is called as hub. Okay, this is called as hub. This is called as hub. Okay. Now students understand. Here we have how many in the periphery? 9 in the periphery. So 9 plus. How many microtubules do you find in the center here? Do you find any microtubules here in the center? Nothing. So can I call it 9 plus 0 arrangement? All of you agree with me? I will call it as 9 plus 0 arrangement. 9 plus 0 arrangement. That's all. This is the structure of centrioles. Not centrosome, actually centriole. A single centriole from the top view. Okay. Now students, if you remember this structure of cilia and flagella, you remember centriole form the basal body of cilia and flagella. Yes, this is the entire flagella. Remember, this was the part is the basal body. So basal body is 9 plus 0. Cilia and flagella on top is 9 plus 2. So basically, this basal body is nothing but same shape as centriole. Basal body. So 9 plus 0 in the basal body. Can you see a triplets? As we go up, it is going to be 9 plus 2 arrangement in the flagella part. Okay. Now all of you remember the function. It helps in cell division. Right? This is during cell division, it helps in spreading out the spindle fibers. Spindle fibers arises. Spindle fibers attached to the kinetochore, and it helps in the separation or formation of metaplastic plate. Okay, function. Now students, now we come to the end of the chapter. Yes, we do. We come to the end of the chapter. Now I'm going to teach you the last topic for this chapter, that is nucleus. Can we start now, students? Can we start nucleus? Can I get a go signal for nucleus now? All of you. Can we start nucleus? Let me just check the chat a little. How is everyone doing? I am from Karnataka. I am also from Karnataka. Same pinch. Right? Same pinch. Who is commenting I'm looking old now? Huh? Who is that? Who is that person who's commenting I'm looking old? Huh? Oi. Who is that commenting person? Some random person I saw, students, I saw it. Went away, I saw. Someone is commenting, sir, you look old. If I look young also problem, if I look old also problem. Amrita, listen. In the case of prokaryotic flagella, we had something called as hook filament, right? We had something called as hook filament and basal body. But here the structure is different, right? Here it is made up of microtubules. Microtubules are absent there. There are no microtubules there, right? There are no microtubules in the case of prokaryote. Prokaryote does not have any of those. Eukaryote will have microtubules. Okay. Same pinch to you also. Nitrosoma, same pinch to you also. If you're from Bangalore, come and meet me also. If you're from Bangalore, students, you're always welcome. You know, right? If you're in Bangalore, you're always welcome to come and meet us at Vedantu Center. Come to HSR layout, meet us. I'll be there. Gopika ma'am is going to be there all the time. So you can meet us also. There is no thing like that. I'll buy you some food. I'll take you to California burrito. I'll give you food also. Don't worry. Okay. We will not send you empty handed. Now students, now we'll start the nucleus. Can we start? Okay. Now can we start nucleus now? Yes, let's start nucleus. Now nucleus is called as the brain of the cell. It's called as the brain of the cell because the entire information, the heredity information, the entire DNA, chromosomes, everything is present in the nucleus. It is present in the nucleus. Okay. It is present in the nucleus. Okay, now let me just keep the phone aside. I get distracted otherwise. Okay. Now the first question to all of you: who discovered nucleus? Can anyone remind me? Who discovered the nucleus? It was Robert Brown. Who has color so fancy, no students? Imagine my name was Basavaraj Red or Basavaraj Orange. So nice. Here we have Robert Brown here. Color is given. Fancy people. Now students, here you need to understand that as nucleus was identified by Robert Brown. But what about chromatin? Chromatin. This chromatin was identified with the help of staining. With the help of staining by a scientist called as Fleming. Again, fancy name, students. Fancy name. This, let me tell you this, Americans, wherever they have nice name. Okay. Now students, can we do the diagram? All this aside, can we do some diagram now? Let's do some diagram and learn now. Let's do some diagram. Let's draw some diagram and learn. All these words you'll understand. Okay. Now let's draw the diagram. That is more important. Okay. Structure you understand. Okay. Let's say this is the center. This is the cell. This is the entire cell. Okay. This is the cell. This is the entire cell. Okay. This is the entire cell. Okay. Santa Claus, I know. Tell me something which I don't know. Okay. This is the cell. Okay. Now in this cell we have the nucleus here. Which color should I take? This became too big. Okay. We have one nucleus here. Okay. This is the nucleus here. This is the nucleus here. We'll take one more. Let's take same color only. It's fine. Perfect. Now students, understand, listen to me very carefully, all of you. Now in this nucleus, right, in this nucleus, here we have something called as outer membrane, something called as outer membrane, yes. Then we have the inner membrane. Then we have the inner membrane. Okay, outer membrane, inner membrane. Now between outer and inner, do you see this middle cap? Here in the middle we have something called as perinuclear space. Perinuclear space. We have perinuclear space here. Okay. Now in this entire structure, right, envelope, there are certain gaps. There are certain gaps here. They are called as nuclear pore. Do you see this, students? All of you see this? Yes, obviously, it's visible. These are called as nuclear pore. Nuclear pore. Now, what is the function of nuclear pore, students? Nuclear pore, right? Why so many things in the chat? What is happening? Okay, nuclear pore. Okay, nuclear pore is the region where multiple things can enter inside the nucleus, like RNA and certain proteins. They can enter and exit the entire nucleus. So entire let's say mRNA entry and exit of mRNA. Done, students? Done? Okay, this is done. Now students, all of you remember, in the nucleus, from the outer envelope we had certain type of organelle, right? Can anyone remind me which is the organelle which is present on the outer surface? This organelle was none other than the ER, endoplasmic reticulum. If it has ribosomes, it is going to become RER. It is going to become RER if it has ribosomes. If it has ribosomes, it becomes RER. Okay. Now you understand, students. This is the outer part. We are done. This is the entire nucleus. Outer part is done now. Okay. Now students, the inner part. Right. Inner part. Now similarly, students, here we have cytoplasm, right? Here we have cytoplasm. Now similar to cytoplasm, inner side also we have some form of matrix. Inside also we have some form of matrix on the inside. Okay, matrix. Okay. And this matrix is called as nucleoplasm. Where do I write it now? Right here. This matrix is called as nucleoplasm. Nucleoplasm. Nucleoplasm is the inner matrix. Nucleoplasm is the inner matrix here. Done? Done. Now students, inside this entire structure of the nucleus, we will find a certain thread-like structures which are completely scattered around everywhere. Okay, see here. Can all of you see this? Scattered everywhere. Yes, or we can see this. Now this structure, I'll write this cytoplasm because all of you know that. This structure here, which is a thread-like structure, which is completely scattered around, that is called as chromatin. That is called as chromatin. Now what is chromatin made up of? Chromatin is made up of your DNA, histone protein, non-histone protein. Remember that. And remember students, if chromatin is visible, this nucleus is right now in interphase. Because it's not dividing, it's not condensation is not done. It is an interphase. It is an interphase. Okay. Now students, inside this structure, there are certain regions. There are certain regions here. Okay. Where there is formation of rRNA. Free floating. Now what are these structures? These structures which are responsible for rRNA, they are called as nucleolus. Where do I write nucleolus now? Okay. This green color one, students, green color one. That is called as nucleolus. That is called as nucleolus. Nucleolus is mainly responsible for rRNA production. rRNA production, nucleolus, rRNA production, and that's it. And that is, student, that's it. What is chromatin? I'll tell you what is chromatin made up of. Don't worry. Okay, this is the entire structure of nucleus, students. Done? No? Tell me, isn't it beautiful? Outer membrane, inner membrane, perinuclear space, nuclear pore, nucleoplasm. Chromatin is present here. Nucleolus is present here, which is responsible for rRNA. That's all. That's all is your entire structure of your nucleus. Okay, entire structure of nucleus is that's all. Okay, nice, no? Students, quickly smash the like button. All of you are new. All the students who are new, quickly smash the like button. Target is 200. Let's go. We have to reach. We have around 41 likes more. Okay. This is what is given in my diagram. Okay. So outer layer, inner layer, nuclear pore. Can you see nuclear pore is here? Nucleolus. This is nucleolus is here. Can you see? Nucleolus is here. Chromatin is here. Chromatin. Okay. This is cistern. Can you press from your Golgi can be also be present here. Golgi can also be present here. Okay. No. Okay. Nucleoplasm, electron microscope. Nucleoplasm here inside. Nucleolus, rRNA. Nuclear pore here. Nuclear membrane. Now students, there are certain cells where nucleus is absent. For example, in the sieve tube plates, in the case of your sieve cells, nucleus is absent. Why? Because for the better transportation. Yes. At the same time, in the case of erythrocytes, that is RBC, nucleus is missing. Why? To accommodate more and more hemoglobin. So it is missing there also. So in certain cases, it is missing. Nucleus is completely absent. Okay. All of you know this, right? This is the chromosome. Chromosome, where if you decondense, you will get long thread like called as chromatin. And this is the DNA. All of you should know this, right? All of you should know this. This is a chromatin. A chromatin will have long chain of DNA plus histone protein as well as non-histone protein. This is a chromatin. So please remember. So what exactly is present in chromatin? Please remember that. A chromatin is basically long thread which has beads. Beads are called as histone protein. That is octamer is present. There is a nucleosome, right? And then we have non-histone proteins can be present here. Okay. Now students, chapter is almost over. Now I'll teach you the what is a chromosome. The structure of a chromosome. Draw with me, all of you. Last diagram for today. Last diagram for today. If you do this, you're good to go. Okay. Last diagram. Okay. Now, for example, if you look at a chromosome, all of you need to know that there is something called as a primary constriction like this. There is something called as a primary constriction. Okay. There is something called as primary constriction. This right here is a primary constriction. This right here is a primary constriction. This primary constriction is called as centromere. It's called as centromere, students. And remember, this is a centromere which is completely inactive in nature. It is completely inactive. Okay, it is mainly made up of heterochromatin. Heterochromatin it is made up of mainly. Okay, inactive in nature. Okay, primary constriction. These are called as chromatids, and these are sister chromatids. Sister chromatids. These are the sister chromatids. Okay, sister chromatids are done. Now, students, around the primary constriction, there is a cap-like structure here. Can all of you see this? Around the primary constriction, there is a cap-like structure where the spindle fibers attach. All the spindle fibers attach to this cap-like structure. This cap-like structure is called as kinetochore. It is called as kinetochore. Kinetochore. Now, this end of them will have this end region, students. This end regions will have secondary constriction. Will have a secondary constriction. This secondary constriction is called as a satellite. Called as satellite. But unlike primary constriction, satellite is going to be active in nature. That is, it can undergo transcription. It is active in nature. Okay. Now this ends here. These ends are called as telomeres. These ends are called as telomere. Are called as telomere. Are called as telomere. And that is the structure of your chromosome. Done. Yes. All of you take a screenshot or write it, draw it, whatever you want. Okay. But smash the like button. Okay. Now here, this part, right, of a chromosome is called as a P arm. The below one is called as a Q arm. This is called as centromere in the middle. And this entire chromosome is basically condensed DNA. Condensed DNA is a chromosome. Okay. Condensed DNA is a chromosome. Okay. There is something called as heterochromatin, euchromatin. Briefly I'll tell you. Euchromatin is a type of chromatin which can be transcribed, which is active in nature. Heterochromatin is very tightly packed and inactive. This is going to be inactive in nature. Inactive. It is very tightly packed. Can you see? It is very tightly packed. Tightly packed. Euchromatin is going to be active chromatin, and it is very lightly or loosely packed. Very loosely packed. Euchromatin. Both of them are present in the chromosome. Okay. Now, based on the position of centromere, there are multiple types of chromosomes. Right. There is something called as metacentric, submetacentric, acrocentric, telocentric. This is the only diagram which you need to understand. Metacentric, the centromere is right in the middle. V shape it can give you. Okay. Submetacentric, the Q arm is smaller. Here we can see a shape that is L shape. Here. Now what about acrocentric, students? Acrocentric, what do you see here? We see almost a J shape. And this is I shape. Telocentric is completely I shape. So telocentric, acrocentric, submeta, metacentric. All of these are different types of chromosomes based on the position of centromere, wherever centromere lies. Okay. This is given your NCERT also. Okay. Now, students, with that, the chapter is over. Done. Now, students, for the next 15 minutes, can we do something called, next 10 minutes, can we do something called as a rapid fire? Can we start? Can we start, students? Rapid fire now. 10 minutes rapid fire. Can we start? 10 minutes rapid fire. If you know the answer, tell the answer in the chat very quickly. Okay. Rapid fire round. Okay. Microbodies are basically small structures which are found both in plants and animal cell. That's all. Don't worry. Okay. Now, students, rapid fire round. I want you to keep a track of your marks. Every single correct answer is going to give you 1 mark. No negative marking. No negative marking. Every right answer 1 mark. Okay. I want quick answers now. Very fast answers in the chat. 3, 2, 1. Let's go. I'm going to be looking at the chat here. Okay. First question on the board. Which are the sites? Which are the following statements concerned about endoplasmic reticulum is incorrect? They're asking incorrect statement. SER are the site for lipid synthesis. RER is as ribosomes attached to ER. SER is devoid of ribosomes. In prokaryotes, only RER is present. What is the answer? Answer number 4. Why, students? Because in the case of prokaryotes, they do not have any ribosomes. Ribosomes are absent there completely. Obviously, ribosomes are absent there. Okay, first question done. Second question. Membrane-bound organelles of endomembrane system coordinates their cellular function. Yes, they coordinate. Mitochondria and chloroplast are not considered part of endomembrane system. Correct. Correct. What is the answer? Both. Answer is both. Marks in the chat. I want everyone to keep a track of your marks in the chat. Keep a track of your marks. Okay. Now, 3. Done. Next question. Match the following. Simple match the following, students. Chromoplast, amyloplast, ileoplast, eleuroplast. Okay. Amyloplast is amylopectin. That is going to be starch. That is going to be starch. So A is going to be 3. Ileoplast is going to be oil. C is going to be 2. Eleuroplast is going to be proteins. D is going to be 1. So A is going to be 4. So A is 4. Right? Done. Done. Answer number 2. Answer number 2. Right? So very easy question again. You all of you know. Here to here. Amylo I told you. Starch. Starch. Eleo oil. Eleuro protein. Done. If I do that, you won't be able to understand. Which type of substance would face difficulty in passing through the cell membrane? Which type of will face difficulty? This is a difficult question. This is a AIMS level question right here on the board. Okay. Substance with hydrophobic moiety, substance with hydrophilic moiety, all substances irrespective of the hydrophobic and hydrophilic moiety, substances which are soluble in lipids, which can find it difficult to pass through. Okay. Here I'll give you a clue. Remember this structure. Polar head, non-polar head, hydrophobic, hydrophilic. Can you tell me? Which will find difficulty in passing through? Obviously, hydrophilic molecules, right? Hydrophilic molecules cannot pass through at all. They might pass through the head, but they cannot go to the non-polar tail. They cannot pass through. Hydrophilic, they cannot pass through. Okay. Here, polar molecules cannot pass through. Next question. Very simple question. Okay. If pH of lysosomes is increased to alkaline, what will be the outcome? That is, all the hydrolytic enzymes will become denatured. They cannot function anymore. Yes. They'll become inactive. Inactive. Simple. Because all the hydrolytic enzymes, they need the pH, acidic pH. They need acidic pH. Next question. Okay. Simple question. Time starts now. Go quickly. Cristae, right? Cristae is short in the case of mitochondria. Thylakoids, they are part of the plastid. Okay. Centromere is the primary constriction. Okay. Cisternae is part of your Golgi complex. Done. Answer is done. That's all. Answer is done. That's all. 1. A is 3. B is going to be 4. Centromere is going to be 1. Say cristae is going to be disc-shaped structure. 1 is the answer. 1 is the answer. You have to be very fast. You have to be very, very fast. Okay. Next question. Which of the following is Incorrect statement. Incorrect. Okay. Perinuclear space formed a barrier between materials present inside of the nucleus and that of the cytoplasm. Okay. Nuclear pore act as a passage for protein and RNA molecules in both directions between nucleus and cytoplasm. Okay. Mature CO tube elements possess conspicuous nucleus and cytoplasmic organelles. Microbodies are present in both plant and animal cells. Students, very simple answer. 3. I told you, mature CO tube cells do not possess the nucleus. Do not possess the nucleus. Organelles that are included in the endomembrane system. Very simple question. Endoplasmic reticulum, no mitochondria, no mitochondria, no mitochondria. So endoplasmic reticulum, Golgi complex, lysosomes, vacuoles. Option number 4. Okay, option number 4, students. Which is the important site for formation of glycoprotein and glycolipids in eukaryotic cell? All of you know the answer. All of you know the answer. Where do we get glycoprotein and glycolipids? Glycoprotein and glycolipids are formed in the Golgi complex. Golgi complex. They present in the Golgi body. Okay. Inclusion bodies of blue, green, purple, and green photosynthetic bacteria. Oh, it's not sulfur, it is photosynthetic bacteria. What do they have? I told you, students, they have the gas vacuoles. They have the gas vacuoles. Next question. Biosynthesis of rRNA. Biosynthesis of rRNA is done where? rRNA. I told you, in the nucleus. Nucleolus has the rRNA. Nucleolus has the rRNA. The concept of omnicell. Okay, answer, students, very easy. Rada Virchow. Doesn't even need to think this one. Rada Virchow. The shorter and the longer arms of submetacentric are. I told students, P arm, Q arm. P arm is a short. P arm is a shorter. Q arm is a longer. P and Q. Today only we learned P and Q. Which of the following pair of organelles does not contain DNA? Does not contain DNA. Okay. Mitochondria contains DNA, gone. Mitochondria contains DNA, gone. Chloroplast contains DNA, gone. Lysosome vacuoles. Lysosome vacuoles do not contain DNA. Do not contain DNA. Which of the following statement is not correct? Lysosomes are found by the process of packaging in the endoplasmic reticulum. Why, students? Why? Because endoplasmic reticulum, it is supposed to be Golgi complex. Supposed to be Golgi complex. And students, all of these are NEET PYQs, NEET, AIPMT as well as AIMS level questions. Not some random questions of the board. Difficult questions. Okay. Lysosome has membrane. All of you know lysosome has membrane, right? Lysosome is part of your endomembrane system. Okay. It is supposed to be Golgi complex. That's all. That's all, students. That's all for today's class. Thank you so much. If you want these notes are going to be available on Telegram channel. Now I want everyone to comment on this video to telling that how was today's lecture, how was the 3D with me, how are the notes with me, and how are the concept learning with me. Tanuja, Endosomes, Navindrani, Bhavana, Ankita, Yashu, Meghana, Christina, Joe, all of you. So I'll be looking forward for your comments. So let me know in the comment section how was today's class. And students, let me tell you, all of these notes will be available in the description. I'll make sure it's going to be available today and tomorrow. By tomorrow, download the notes and study. Okay. So bye bye, students. Take care. Have a great night ahead. And thank you so much. Bye bye, students. Boy, all of you, amazing. Bye.